So I'll do chemical analysis of plant tissue. For that I have taken the coriander leaf. Now I am putting this into the pestle and mortar to make a proper paste of this plant. Hey, what happened?
Nothing, sir, nothing.
So this is the plant tissue paste. Now what you have to do, you have to make a thick slurry of this by adding trichloroacetic acid.
Wow, this green chutney is so delicious.
Wait. That's not a green chutney. That's basically a thick slurry. By adding trichloroacetic acid, will prepare it the process of analyzing biomolecules. Yes, I'm talking about the chapter Biomolecules from class 11. So don't think that it's a boring chapter, because here we learn with fun at PW NEET English. Myself Biswajit, your zoology mentor. And obviously after completion of this chapter, we'll practice a lot of questions, and for that you must have to click the link given in the description box, which is gateway to PW app. The most amazing fact about living organisms is that they're entirely made up of non-living molecules, and we call them biomolecules. Every day we are eating food, and these foods are laden or rich with nutrients. We call them carbs, we call them proteins, we call them fats, we call them vitamins and minerals. Okay, now when I'm talking about carbohydrate, protein, fats, all of them falls under the category of biomolecules. Few are larger in size, few are smaller in size. So in this chapter we'll clear each and every confusion and concept of this chapter. So these are the topics we are gonna cover for this chapter. So let's start one by one. Okay, so first we'll start with the definition of biomolecules. So biomolecule means, firstly, all of the biomolecules are organic in nature. Now what do you mean by organic? Organic means a molecule which has carbon and hydrogen atoms inside that. So a molecule or compound of carbon and hydrogen are called organic compounds, and biomolecules are organic in nature. Like for one of the example I am writing here, when you talk about glucose, C6H12O6. So you can find here glucose is made up of carbon, hydrogen and oxygen. So that's why we can call it a biomolecule. And obviously it's written in NCERT that all carbon compounds present in a cell are also can be called as biomolecules. So obviously these carbon compounds are organic in nature. These carbon compounds are organic in nature. So basically we have to peep inside the cell, and we have to analyze the molecular analogy, the molecular differences, the molecular variation inside. Okay, so when you are talking about biomolecules, there are of two types. First, biomacromolecules and biomicromolecules. Now by the name you can understand micro means small and macro means large. Okay, so which biomolecules are called biomacromolecules? The molecules or the biomolecules which has a molecular weight of more than 1000 Dalton, and usually they are even larger than 10,000 Dalton. Yes, this is the unit of molecular weight. Now what about biomicromolecules? The biomolecules whose weight is in between 18 to 800 Daltons we measure, we call those molecules as biomicromolecules, because they're obviously smaller than biomacromolecules. Okay, in detail obviously we'll study about them. Now let's move forward. Now we'll start with one of the important topic of this chapter, that is elemental analysis. Now we'll check for elements present in different types of living being, and we also compare the elements present in our body, present in living being, and elements present in earth crust. because whatever we are eating every day, we are inhaling every day, everything we are getting from this environment, from this earth, right? We know that our body is made up of panch tattva, right? Now that's why is the reason we need to compare between the elements or the components of living being and non-living being, because the food every day we are eating, they are made up of nutrients, and nutrients are obviously non-living material. Okay, so first we will take a piece of plant tissue. You can take any young leaf of a plant, or else you can take animal tissue, piece of a goat liver, or else you can take microbial paste. Now what we have to do here, what we will do, we will just send the tissue paste into chemical lab for their elemental analysis, and the chemist will tell us that the plant tissue has these sorts of elements present into that. Who are they? They are carbon, hydrogen, oxygen, phosphorus, nitrogen and sulfur. You can remember them in this manner, chopans. So these are the major elements present into plant tissue, and there are also a few minor elements you will be finding inside them. They are pronounced as the NaCa-Si-Mag. So sodium, calcium, silicon and magnesium. Okay. Now let's talk about the elemental analysis of animal tissue. That goat liver you had taken, now you have sent it to the lab for chemical or elemental analysis, and the chemist said or given you the same report. Even the animal tissue paste that is made up of similar kind of elements like carbon, hydrogen, oxygen, phosphorus, nitrogen, sulfur, sodium, calcium, silicon, magnesium, chopans, NaCa, Magsi. Okay, now you have taken microbial paste. So you thought that these microbes means they are mostly prokaryotes in nature. You have taken a bacterial culture. Now you have sent them to the chemist for the elemental analysis, and surprisingly you got the similar kind of result. Even the bacterial paste that is also made up of chopans, NaCa, Magsi, right? Okay, so you have concluded that though living organisms, they are highly diversified, but they have very basic similarities in between them, because the diversity when this evolution had started from a single point. So that's why they have a similar setup of elements, but it's possible that in few organisms a particular element present in higher amount, and in another organism a particular element present in lower amount. Okay. Now you have to do, that is the analysis or elemental analysis of earth crust. So what you have to do now, you have to take one piece of stone or one piece of brick, or you can take a handful of mud. So that is called piece of earth crust or part of earth crust. Now you will do the same thing, you will make a paste or powder of it, and you will send to the chemistry lab for the elemental analysis. And here again surprisingly you got the similar kind of result. The earth crust even made up of similar kind of elements, chopans, NaCa, Magsi. So here you can conclude with this sentence that all living and non-living beings are made by similar elements, but they differ in relative abundance. So that is the concluding statement after doing the elemental analysis. So you can say this, obviously same but different, same but different. Let's move to the next slide. So here what we have done, C.N.R. Rao, one renowned biochemist, he observed, he studied the elements present in earth crust and elements present in our body tissue. Okay. So here we will do a comparative analysis of elements. in between earth crust and human body, and we will also try to find out that what kind of important questions can be framed from this particular table given in NCERT. Okay. So, as you know that carbon, hydrogen, oxygen, nitrogen and sulfur, they are the major elements present into living beings. Sodium, calcium, silicon, magnesium, they are the minor elements present into living being. So, let us do a comparative analysis. So, for carbon, you can observe here that earth crust has 0.03 percentage of carbon, whereas human body has quite high percentage of carbon inside us, that is 18.5%. Okay. So, in earth crust, when you are talking about the coal, right, it's a huge reservoir of coal, the coal mines, then you have to talk about the carbon in earth crust. Now, when we are talking about human body or body, basically suppose you are 18 years old. Now, within this span of 18 years, you have eaten 1000 kilograms of food, right? And the food materials, the nutrients are again carbohydrate, proteins, fats. Now, the neve material, the main backbone of all the food components, they are basically hydrocarbons. They are hydrocarbons. So, basically when you are talking about glucose, remember it has a formula of C6H12 and O6, right. So, that C carbon, every food materials or nutrients every day we are taking and we are digesting, and they are helping us to build our body, build our bone, build our muscles and everything for our physiological functions. They are helping our physiological functions. So, carbon in present in carbohydrate, carbon in present in proteins, carbon in is present into fats or lipids. So, that is why we can say that very high percentage of carbon is present into our body, and even we burn, when we will burn living being, when we are burning a living being, we will be getting some ash. So, when we are converting a living body into ash, so we are doing combustion or oxidation, and that is producing large amount of carbon dioxide. Okay. Now, let us talk about hydrogen. Okay. So, in earth crust, hydrogen is very present in a very minimal percentage, 0.14%, but in case of human body, the presence of hydrogen is again quite higher, 9.5%. Now, when you talk about oxygen, the highest percentage both in earth crust and human body, in earth crust oxygen is present as 46.6%, whereas in human body again oxygen is present. The percentage of oxygen is 65.0%. Oxygen is dissolved into our blood. Oxygen is present as important element of carbohydrate, proteins, fats, and even DNA and RNA. Okay. Now, when you are talking about nitrogen, earth crust has a very negligible or little amount of nitrogen into it, because nitrogen, most of the nitrogens, the 78% nitrogen is present into this atmosphere, and rarely they get stored inside the soil. So, that is why a very little amount of nitrogen is present in earth crust, but when you are talking about human body, 3.3% is quite higher a percentage of nitrogen present into us, because when you are talking about protein, particularly protein, when you are talking about body growth and development, body building nutrient protein, that needs nitrogen for its core component, right. Without nitrogen you cannot make protein inside your body. Yes. Now, let us talk about sulfur. When you are talking about sulfur, in earth crust the percentage is 0.03%, and in human body the percentage of sulfur is 0.3%. Now, if you compare all these, elements, you will find that for every case, carbon, hydrogen, oxygen, nitrogen and sulfur, their abundance is much higher in case of human body, but lower in case of earth crust. Now, when we will observe the abundancy of this particular elements, sodium, calcium, silicon and magnesium, the condition is reverse. You can check it here. The earth crust, when you talk about earth crust, the percentage of sodium is 2.8%, but in case of our body, the percentage of sodium is only 0.2%. When you talk about calcium, so calcium in earth crust 3.6%, in case of our body is only 1.5%. Calcium, again, earth is a reservoir of calcium in form of different stones like marbles, right? In our body, the reservoir of calcium is basically our bones and teeth. Okay. When you are talking about silicon, again, earth crust is a huge reservoir of silicon. It has 27.7% of silicon, whereas in case of most of the living being, silicon is toxic. So that's why it's very rare or negligible in case of living organisms, but in case of human body. But few porifera, few sponges, they have siliceous spicule inside their body. Okay. Now come to the magnesium. In earth crust you will find 2.1% of magnesium, but in case of us, in case of our body, is only 0.1%. Okay. Now that was the comparative analysis. Now let's talk about the questions. How can you form, how can you frame questions from this table? So if you arrange the relative abundancy of elements in earth crust, then what kind of pattern you are gonna get? The first rank is of oxygen, right, 46.6%. Okay. The second rank is silicon, 27.7%, and 46.6, 27.7. The third rank is of calcium, and the fourth rank is of sodium, 2.8. Yes. So this is the pattern of abundancy of elements in case of earth crust. Now let's talk about human body. Again, the highest abundancy is for oxygen, 65.0%. Then the second highest abundant element in case of human body is carbon, 18.5%. Third highest, hydrogen, 9.5%. There is a data correction in old NCERT. You will get the correct data in your new NCERT regarding this hydrogen. Okay. And the fourth abundant element for human body, that will be nitrogen. Okay. So question can be asked in this manner, right? Now question can be asked that an element which is very little in earth crust, but which is the fourth most abundant element in case of human body. Here we have to talk about nitrogen. Okay. Another question can be asked, an element which is present in negligible amount in human body, but which is the second most abundant element in case of earth crust. Here we have to talk about silicon. So this kind of questions can be asked, right? So do not forget to study this table given by C.N.R. Rao in a detailed manner, in searching oriented mindset, right? So you need to remember about nitrogen, you have to talk about silicon, right? Because nitrogen is very little in earth crust but is abundant in human body. Silicon, opposite case, highly abundant in earth crust but very little in case of human body. So in this manner you can memorize them. Okay. Now let's move forward. So as I was telling that, why I have given a barrier here, this elements versus this elements. So the reason is that in our body, for living being, chopons or chons, in NCERT they haven't talked about phosphorus, carbon, hydrogen, oxygen, nitrogen, sulfur, they are regarded as the major elements. Almost in every food you are going to get them. Carbon is present in all the nutrients, hydrogen is present in all the nutrients, oxygen as well as present in all the nutrients, nitrogen special element in protein, sulfur special element in few types of amino acids like cysteine, methionine. They are building blocks of protein, you will get sulfur, even you will get sulfur in the building matrix of our cartilage, it is chondroitin sulfate. Okay. And obviously when you talk about sodium, calcium, magnesium, silicon, they are present in quite a low percentage in our body if you compare them with these other elements. So that's why we call them minor elements. Sodium, where do you need sodium into your body? Sodium potassium pumps are very much important, you know. So they are acting as major electrolytes. You will present sodium in extracellular fluid into our body. Calcium, they are present into bone, they are present into teeth, they are also acting as electrolyte. Calcium, they are acting as clotting factor, help in clotting, acting as clotting factor. They also help some enzyme for their activities. Okay. Calcium also help in nerve impulse propagation, nerve impulse transmission. Calcium helps in the process of nerve impulse transmission. Magnesium also helps some enzymes for their activity. And as I said, silicon is very rare in human body, but obviously is present in sponges as siliceous spicules, inside siliceous spicules. The siliceous spicules are there, a non-living endoskeleton. Okay. Now we will talk about one experimental study. You can take any of the living tissue, either you can take a plant, you can take any leaf, or you can take any animal tissue, like you can bring a piece of goat liver. Now you have to grind it, either it is plant or animal, and after grinding we will add a special solvent, polar solvent into that, and that is trichloroacetic acid, and after adding that we will separate out few components from them by the process of filtration, and we will study for the presence of biomolecules as well as inorganic components in different fraction. So that process is called chemical analysis. Chemical analysis, and why we are doing chemical analysis? Mainly our aim is to find biomolecules present into our cells or tissue. So for our experiment here we have taken a living tissue in the form of a leaf. So you have taken a leaf, so you have taken leaf or leaves. Next thing we will be grinding them, and how you will grind them? You will grind them with the help of pestle and mortar. You have observed in your kitchen. If you haven't observed, ask your mom, please give me pestle and mortar, and by utilizing this machine we will make a proper paste of this plant leaf. And next what you have to do, you have to mix tissue paste with trichloroacetic acid. Remember the formula of trichloroacetic acid, that is Cl3CCOOH. Okay. Here you can observe the structure of trichloroacetic acid. So basically in the middle there is a carbon atom and it has 3 chlorine atoms associated with this. That's why we are calling it trichloro, and here you will get carboxylic group. So that's why we are calling it trichloroacetic acid. Now after adding this, we will make a thick slurry inside a glass jar. Now what you have to do, you have to filter it out or strain through using cheesecloth or cotton cloth. Now we will get 2 fraction, one is called filtrate and one is called retentate. Filtrate also can be called acid soluble pool, retentate also can be called as acid insoluble pool. Now here you can observe how this experiment is done by when you are putting this thick slurry. When you are putting this thick slurry into the cheesecloth or cotton cloth, and you have to put pressure, that is called process of straining, the liquid portion or the acid soluble portion will come down into this glass container, and we will be calling it filtrate, and the contents which were unable to dissolve in the acid which is present inside the cheesecloth or cotton cloth, we will be calling it retentate. And remember that this filtrate are polar in nature, and this retentate, the molecules are mostly non-polar in nature. Now with the help of a live experiment, we will see how this process of chemical analysis is done. So why only theories? Let's learn the concept of chemical analysis from the chapter of biomolecules with proper practical experiment. So for that we need plant tissue paste. So I have collected coriander leaf. Next we need a pestle and mortar. So I am putting the coriander leaf. Now we will make a proper paste of this. Now when this paste of coriander leaf is ready, so I have to put it into another glass beaker. So I am transferring it. Now as because this is too much thick to make a homogeneous mixture, I will add trichloroacetic acid into this. So a little bit of trichloroacetic acid. Now this will make a thick slurry, right? Now I will put this into a cheesecloth to squeeze out the liquid part. So let's perform that. So for that I have taken a glass jar. This is the cheesecloth. I am putting thick slurry on it. Okay. Now as it is said in NCERT, I have to strain the cloth. You can observe here green colored liquid is coming out when I am putting pressure. I am straining the thick slurry. So we got 2 different fraction. Okay. So the liquid portion which is present inside this glass jar, that is called filtrate, and the acid insoluble fraction that is trapped within the cheesecloth, that is called retentate. Now we have to perform proper chemical analysis. We will try to find what kind of biomolecules are present into this retentate and what kind of biomolecules are present into this filtrate. Okay. So what we got? filtrate and retentate, filtrate and retentate. So when we are doing that experiment, basically we had taken leaves or leaf. When we grinded it, basically we were crushing the cells present into it, and after grinding we had added trichloroacetic acid, Cl3CCOOH, trichloroacetic acid, to make a homogeneous mixture, as we have observed. Okay, so basically when you make chutney in your home, with that coriander leaf paste, you add simple water. So basically that's a universal solvent. So here as solvent we have used a polar solvent, that is trichloroacetic acid. Okay, and we got two fraction. One is filtrate. Now what about this filtrate? The filtrate has all the smaller and polar cytoplasmic components, because you know that this trichloroacetic acid, that is a polar solvent. Trichloroacetic acid, that is a polar solvent. Trichloroacetic acid is a polar solvent. And polar solvent will dissolve polar molecules. Polar solvent will not be dissolving non-polar solutes, right? So with this acid, smaller and polar solutes or cytoplasmic components will come down and get stored inside that glass jar. So we are calling it filtrate. Also we are naming it acid soluble pool. Polar dissolves, polar solvent dissolves polar solute. Okay, but when you are talking about retentate, all larger and mostly non-polar cytoplasmic, cell organelles, cell membrane, cell wall and nucleus components will remain inside the cheesecloth, because polar solvent is unable to dissolve non-polar solutes. So that's why the retentate is also called as acid insoluble fraction. So very important questions can be asked from this. Like if I tell you that you have observed that all smaller and polar cytoplasmic components are present into filtrate. So if you get a statement, all polar cytoplasmic components are present into filtrate, that is the true statement. If you get a statement that all cytoplasmic components are present into filtrate, so that will be a false statement. Okay. Now what kind of cellular fraction you are going to get inside that retentate? You will get non-polar cytoplasmic components. So when you are talking about cytoplasmic components, polar cytoplasmic components are present into filtrate, whereas non-polar cytoplasmic components present into retentate, right? And obviously the parts of cell organelles, cell membrane, cell wall and nucleus, as because they are larger in size, they will be present into retentate. So that's how you can differentiate in between the cellular fraction which are present into filtrate, which are present into retentate, right? Okay. Now we'll make a we have made a differentiating table between filtrate and retentate. Now while you are studying this, please do double check your learning outcome of filtrate and retentate in different way. In a two-step manner, statement based questions can be given, right? So what are the components present in filtrate? Obviously mostly they are monomeric in nature. Like when you talk about amino acids, when you are monosaccharides, when you're talking about nucleotides, nucleosides and nitrogen bases, all of them are monomers. Now, if you don't understand monomer, if you have a garland or a ring with beads, now if you unchain it, you will find, will separate out these flowers from that garland. So this long chain can be called as polymer, and these separated beads can be called as monomer. Now to form polymer, you need help of monomers. So the components present in the retentate, you will get proteins, polysaccharide and nucleic acid. These three, you can call them polymer or polymeric substance. Like proteins are polymer of amino acids, polysaccharides are polymer of monosaccharides, nucleic acids are polymer of nucleotides, right? But in acid insoluble fraction or within retentate, you will get an imposter. You will get an imposter. They are lipids. That's why I have written it into a different color. Why I am telling that lipids are imposter molecule mixed with these polymeric substance? Because lipid is a non-polymer. Lipid is a non-polymer. Okay. So it's written in NCERT, it's not strictly polymer and not even a biomacromolecule. Even these proteins, polysaccharide and nucleic acids, even they are biomacromolecule. Proteins, polysaccharide and nucleic acids, mostly they have size more than 10,000 Dalton. But when you are talking about lipid, when I am talking about lipid, the molecular weight is less than 800 Dalton. So technically lipids are biomicromolecule, but present within the mixture of biomacromolecules with proteins, polysaccharide and nucleic acids. Now what is the reason? Because lipids are non-polar in nature. Lipids are non-polar in nature, but we know that filtrate has all the polar components. Now here inside the filtrates, you will get some inorganic components as well. So filtrate has organic as well as inorganic components, whereas retentate has most of the organic components. So what kind of inorganic components you are going to get inside the filtrate? You are going to get sodium ion, calcium ion, potassium ion, chloride ion, sulfate ion, phosphate ion, even water and protons. Okay. So I was talking about lipids. So let's elaborate it. So I said that lipids are polar molecule, lipids are non-polar molecule, lipids are hydrophobic molecule. So hydrophobic means phobia of water. So water is a kind of polar solvent, that's why lipid molecules will not get dissolved into polar solvent. Here we are not, though you are not using water, we have used trichloroacetic acid. So lipid is, as because it's a non-polar and hydrophobic, it won't make friendship with another polar solvent, even it's trichloroacetic acid. So what kind of property you can observe in lipid? So you can take one glass of water, and you have to put few drops of oil upon it. Now what you can observe, even you are shaking the water after putting the drops of oil into the water, if you shake it, even after that as well, what you can observe, the lipid molecules or the oil molecules will not get dissolved or mixed with water, it will remain floated on the upper surface. There will be a proper separation boundary in between oil molecules and water molecules. So basically that property is called aggregation property. That property is called aggregation property as because they are hydrophobic. This is called hydrophobia. So what do you mean by aggregation property? Aggregation property means the property of formation of lipid vesicles. So what you can do, you can take a water pool, you can take a pool of water, this is the pool of water, and after that you have to put few drops of oil into that. Now after some time what you are gonna observe, all these drops are coming together by some attraction force and they will form a large oil globule, oil vesicle. So that is called the aggregation property. So here the same thing is said here. If lipid molecules are closer to each other, they will generate one attraction force towards each other, that is called lipophilia. Lipid molecules are lipophilic but hydrophobic. So due to this lipophilia, the attraction force towards one lipid molecules to another lipid molecule, they will try to attract each other and gradually they will form a large lipid vesicle. So that's why though they are not biomacromolecule, they are biomicromolecule, but sometimes they behaves like they are biomacro because of this aggregation properties. That's why they are present inside the written text. Okay. Now we have come to the slide where we are going to talk about the difference between biomacromolecule and biomicromolecule. We were talking about filtrate and retentate. Now we will talk about micro and macromolecule, and we will try to differentiate between them with proper concept. So this is actually very easy thing. When you talk about biomacromolecule, how we can differentiate by observing their molecular weight. So for biomacromolecule, the molecular weight is more than obviously more than 1000 Dalton, and if you get a statement the molecules which has molecular weight greater than 800 Dalton, even greater than 800 Dalton, even we will be considering them as well as biomicromolecules. So the examples are proteins, polysaccharides and nucleic acid, but you know that most of them even larger molecular weight, they have larger molecular weight than greater than 10,000 Dalton. But let's come to biomicromolecule. When you are talking about biomicromolecule, their weight, molecular weight will be in between 18 to 800 Daltons, and their examples are amino acids, monosaccharides, nucleotides, nucleosides, nitrogenous bases. All of them are component of nucleic acids, and obviously the imposter one, that is lipids. Lipids are also considered as biomicromolecule. Okay, so we are done with how to analyze the biomolecules, either they are macro or micro present into cells or tissue. Now we will try to analyze the process, how we can obtain or separate out the inorganic components present inside living being. Okay. Now we were talking about organic components. Now we will find out a mechanism to extract inorganic components from cells or tissue. So basically here we have to perform a destructive process. We have to perform a destructive process. That means we have to perform combustion, and the process is called as ash analysis. It's not that ash what you are doing with your father's cash. This is ash analysis. For that, again you need living tissue. Again you need a piece of living tissue. Again you have taken one live leaf, any young leaf from any plant you can take. You have to sun dry it. You have to sun dry it, and after sun drying, what we will do? The sun drying will help in the process of evaporation of water present into this. So when you have taken that alive tissue, the weight, we call it wet weight or the normal weight, right, the physiological weight. Now after this evaporation, after the process of drying, the weight you are going to get, we will call it dry weight. So you can observe, this is a dry leaf upon sun drying. Now you have to perform the destructive process, that you have to burn it with fire. You have to burn it with fire, and after this process, basically when it's getting burned, it's getting oxidized. It's getting oxidized, and the hydrocarbons present inside that, the organic compounds, will get evaporated by forming smoke of carbon dioxide and water vapor. And the remaining part, that is called ash, that is laden with inorganic components. And the inorganic components inside this ash you are going to get, except water. Water is also inorganic, water present into our body. So because the water is evaporated, the inorganic components you are going to get inside the ash, that is sodium ion, potassium ion, calcium ion, magnesium ion, bicarbonate ion, HCO3 minus, sodium chloride, salt, calcium carbonate, CaCO3, don't call it CaCO3, phosphate ion, PO4 3 minus, sulfate ion, SO4 2 minus. They are also part of negatively charged protein of a cell. Okay, so that was all about the analysis of inorganic components by the method of ash analysis. Okay, so now we will try to find out the average components present inside our every cell. Okay, so the data is again taken from our NCERT book as it is. So it's like this. See, what we call what everywhere, it's the new year, and you can listen the song Blue Hai Pani Pani. So 70 to 90% of our body weight, of our physiological weight or wet weight, is of water. Basically, if you evaporate it, you will become ash. So that's why you need to remember 70 to 90% of our body weight is only water. Now water is called pani, so the second most abundant. So water is ranked first. The second most abundant biomolecule, that is protein. Water for pani, P for protein. Now if you pronounce protein like this, protein, so it's ending with 10. So the abundance of protein into our body is 10 to 15%. 10 to 15%, protein to 10 to 15%, right? Okay. Now after protein, we will get nucleic acid, the third most abundant molecule present into our cell. Now protein, if you spell it P-R-O-T-E-I-N, it's ending with N. So that's why from here you can configure a trick. The third most abundant is nucleic acid. Protein end with ends with N, not proteins, protein ends with N, and nucleic acid start with N. Now, what percentage of nucleic acid present into our cell? If you count them, acid 1, 2, 3, 4, 5 letters are there, nucleic 1, 2, 3, 4, 5, 6, 7 letters are there. So, it is written as it is, nucleic acids are present into our cell, that is of 5 to 7%, right. Okay. Now, after nucleic acid, when you are pronouncing nucleic, you are giving emphasizing on click C. So, the fourth most abundant molecule into our cell, they are carbohydrates. Now, what percent of carbohydrate? See, when you had studied the English alphabet, first it's come A, then B, then C. So, it's the third letter of English alphabet. So, it's present, the abundance will be 3%. Okay. Now, after carbohydrate, it's lipid, the fifth abundant cellular component. Now, what percentage of lipid is present into our cell? If you observe, if you see this spelling of lipid, there are use of I for 2 times, LIPI. So, 2 I's. So, this is the trick to remember the percentage of lipid, that is why 2%, and the last one, that is ion. Ion, how many I? Only 1 I. So, ions are present, the abundance will be 1%. So, that is the trick. So, again many questions or test statements can be asked from this table as well. If as a whole you were asked that make a chronology of the relative abundancy of components of cell, so it will be like this. First it will be water, then it will be protein, then it will be nucleic acid, then it will be carbohydrate, right. So, the most abundant component of cell is water, but you can the examiner can twist this question in this manner, which is the most abundant, organic cellular component. So, the most abundant organic cellular component will not be water, because water is inorganic. So, our answer here will be protein. Most abundant organic component. Most abundant organic component in cell is protein. Most abundant component, only most abundant component is water. Let us move forward. Okay. Now, one by one we will talk about the monomers and polymers in terms of biomolecular discussion. So, if we are talking about proteins, the monomers, proteins are polymers and the monomers of proteins are amino acids. Proteins are polymers and their monomers are amino acids. So, we will start our discussion with the study of amino acids. Okay. So, few basic information about amino acids. They are biomicromolecules, they have weight less than 800 Daltons, they are monomer of proteins. We also call them substituted methane. Now, why we call them substituted methane? So, the formula of methane is CH4, and this is the molecular structure, a central carbon atom, and it has 4 valent arm, and with each arm you will get hydrogen. Now, what you have to do? You have to substitute this hydrogen of methane with few other groups. So, we will keep one hydrogen as it is, but for rest 3 we will substitute them, one with amino group, one with carboxyl group, and another with R group, also it is called as alkyl group. So now it's a perfect substituted methane. So that's why amino acids are called substituted methane, because substitution of 3 hydrogen atoms are done with amino group, alkyl group and carboxyl group, right? So that's why, as because this molecule, the substituted methane, has one amino group which is basic in nature, and this carboxylic group which is acidic in nature, we call this as amino acid. We call this as amino acid. Now one more thing is written here, all the amino acids are alpha amino acids, all the amino acids are alpha amino acids. Now what do you mean by alpha amino acids? Yes, the carbon present here, that is called alpha carbon. That is called alpha carbon. Now how you can define this alpha carbon? The carbon which is associated with this carboxyl group, COH group, that carbon is called alpha carbon. Now as because this is the basic structural formula of all the amino acids, so each and every amino acid, for each and every amino acid you will get this alpha carbon. So all amino acids are called alpha amino acid. Again I am repeating, the central carbon of amino acid which is associated with carboxyl group, the carbon which is associated just next to this carboxyl group, that is called alpha carbon. Here is the central carbon atom. Yes. Okay. Now if you observe it very carefully, this carbon atom, in one valent position it has hydrogen, in another valent position it has amino group, in another valent position it has alkyl group. You can put a methane, you can put here hydroxy methyl group, and in the fourth valent position you will get always carboxyl group. So a carbon with its 4 hand holding 4 different groups, so that's why this carbon is also can be called as chiral carbon. So most of the amino acid will observe optical activity, right? We'll discuss about that. So what we got to know, all the amino acids are substituted methane and they are called alpha amino acids, and the 4 groups which are associated with each and every amino acids, the 4 groups, they are hydrogen, amino group which is acidic, COH carboxylic group, R or alkyl group, and again for most of the amino acids, for all the amino acids, amino group, hydrogen group and carboxylic group will be constant, but in nature there are more than 500 amino acids you will be getting into living organisms, and what makes difference among them, that is the R group or alkyl group. So that's why we are telling that R group is the variable group of amino acid. H, NH2 and COH, these are the constant group we will be finding in all amino acid, but the alkyl group is different in case of different amino acids. Like for few amino acids the R group is only H, instead of R there will be only another H group. For one amino acid the R group can be CH3. For amino acid another amino acid the R group can be CH2OH, that is hydroxymethyl group. For another amino acid the R group can be CH2SH, thiol group. So you can see here for different amino acids there will be change in the position of this R. So that's why it's a variable group. This is the variable group into different amino acids. Okay. Now let's talk about some examples given in our NCERT. How the variation of R group will cause variation in among amino acids. So what is the generalized formula for R group is CnH2n plus 1. So basically, if you observe this particular group, CH3 methyl group is following the formula CnH2n plus 1. Here C is 1 and H2n plus 1 is 3. So it's satisfying, right? So when you talk about the simplest amino acid, in case of R group, whenever you got H, so that amino acid is called glycine. Here instead of R, you will get H group. We will talk about this glycine. This is an exceptional amino acid, because here if you observe the central carbon of glycine, the 4 different valent position is not occupied by 4 different groups. Here you will get 2 similar groups here for 2 different valent position, that is hydrogen. That is why we cannot call this carbon as chiral carbon. So glycine, for glycine the carbon is, for glycine, only for this glycine the carbon is not a chiral carbon, not a chiral carbon. So that is why glycine won't be exhibiting optical isomerism. Okay. Now if you replace the R group with methyl group, then you will get this amino acid alanine. If you replace the R group with hydroxymethyl group CH2OH, you will get the amino acid serine. If you replace the R group with thiol group CH2SH, you will get the amino acid cysteine. Okay. So few more important info about amino acid. So except glycine, what I said, except glycine, all amino acids have chiral carbon. I said the reason, and optical activity. What do you mean by optical activity? Dextrorotatory and levorotatory form. We call it D form and L form. So suppose this is a pen, I am holding it with my right hand. So this is my dextrorotatory form. Now if I rotate my body, then what will, from your perspective of view, you will observe that my left hand is picking this pen. So that is called my levorotatory form, right? So basically they are mirror image of one another. They are mirror image of one another. So with one easy example, I am showing you. So if this is our friend Chintu, and with his hand he is holding something, holding a ball. Now suppose this is the left hand, this is the right hand. So with left hand Chintu is holding the ball, so we will be calling it as L form of Chintu. Now again the same Chintu in mirror image, he is holding the same ball into the opposite hand. Into which hand? Into right hand now. Now this form of Chintu, we will call it as a D form of Chintu. So that is also applicable in case of molecular arrangement, right? So now you got what is D form and L form. Now why glycine is not qualifying this? Because in case of glycine, suppose, so this is the normal optically active Chintu. This is normal optical activity, optical isomerism. But in case of optically inactive, so this is Chintu's friend Mintu. So with both the hand, he is holding two similar looking football, right? So this is the left hand, suppose this is the right hand, this is the left hand. But what will happen if you make a mirror image? So if you make a mirror image of Chintu, you will get the similar kind of result. The hands will be changed from left to right and right to left, but he will look similar in both the poses. So we can say this Chintu's friend Mintu is optically inactive, because when I was holding one pen with my one hand, my another hand is empty. Now if I exchange the position, it will make a different configuration. But if I don't have any pen in my hand, so what will say, if I exchange my right hand and left hand, so the both of them will look similar to each other. So then I will become optically inactive. So that is the case of glycine. Okay. Now let's move forward. So glycine is the simplest among the amino acid, because the R group for glycine is basically hydrogen. Okay. Now if you ask, sir, what is the most complex amino acid? So the answer will be tryptophan. So the structure of tryptophan is given here. This is the structure of tryptophan. You can observe it's quite complex amino acid. Okay. Now we will talk about only 20 amino acids, though there are more than 500 amino acids present into nature, but we will talk about only 20 amino acids, because these are amino acids which we require for our cell, for protein synthesis, for the process of translation, and these amino acids are called proteinogenic amino acids. Okay. Now next thing, a very important statement based question can be asked: physical and chemical property of amino acids depends upon. Now if most of the students will make a mistake here, we will think that as because R group is the variable group, that is the only group responsible for a variation in physical and chemical property of amino acid, but it is clearly written in NCERT. The physical and chemical property of amino acids depends upon three of their groups, depends upon amino group, depends upon carboxyl group, as well as depend upon R group. How? Let's check into the next slide. Okay. So here you can observe for both the amino acid, the R group is different, right? So here this is the R group, for this amino acid this is the R group. Now what's similar in between them? Each has these constant groups, one amino, one hydrogen, and one carboxylic group. Okay. Now I am drawing another amino acid here. Okay. So this is our alanine amino acid, you know it very well. So here you can observe, for this particular amino acid you will get one basic group, NH2 amino group is the basic group, and carboxylic group is the acidic group. So as because it has one basic group and one acidic group, the basic group will neutralize this acid, and that will make this amino acid a neutral one. That will make this amino acid a neutral one. But for this particular amino acid, you have to calculate how many acidic group and how many basic group is or are present into this. Okay. So here one basic group, one acidic group. Here with this R group we will get another acidic group is present. another acidic group is present. So overall for this amino acid there are two acidic group and one basic group. So overall who wins? Overall acid wins. So you can call this amino acid is one of the acidic amino acid, AA for amino acid. Now when you are talking about this particular one, here one basic group, one acid group, and here another basic group. Now for particular this one there are two basic groups are there and one acidic group is here. So who will win here? In case of this particular one, basic group will win. So we can say this amino acid is basic in nature. So whichever amino acid is given as an example in front of you, and if you are asked that identify is this a basic amino acid or acidic amino acid, what you have to do? You have to calculate the difference between the acidic and basic group. Which is acidic group in amino acid? Carboxylic group. Which is basic group in amino acid? Amino group. So if there is similar number of amino and carboxylic group, there will be neutral amino acid. If there is more carboxylic group, then that will be acidic amino acid. If there is more number of amino group, that will become a basic amino acid. Okay. So based upon that, either is basic, acidic or neutral. Amino acids are mainly of three types: acidic amino acid, basic amino acid and neutral amino acid. So the examples of acidic amino acids are given here. They are glutamic acid and aspartic acid. Only two acidic amino acids are there. Now when you talk about basic amino acids, there are three basic amino acids, only three: arginine, histidine and lysine. Arginine, histidine and lysine. Now if you exclude this 5 out, you know about 20 amino acids. 2 acidic, glutamic acid, aspartic acid. 3 basic, arginine, histidine and lysine. Now if you exclude this 5, now rest 15, all the amino acids are neutral in nature. Rest 15, all the amino acids are neutral in nature. Few examples here, like valine, glycine, alanine, serine, they are example of neutral amino acid. Okay. Now there is a special group of neutral amino acids, we call them aromatic amino acids, and what you have to check, if you will find that the R group has this benzene ring kind of structure associated with R group in a particular amino acid, then that neutral amino acid will become aromatic one. That neutral amino acid will become aromatic. Aroma means smell, the amino acid which are responsible for smell. Okay. So the examples are given here: tyrosine, phenylalanine and tryptophan. Tyrosine, phenylalanine and tryptophan. So here you can observe, this is the structure of phenylalanine, this is the structure of phenylalanine, and here you will get this aromatic benzene ring. Okay. Now if you add one hydroxyl group here, OH, then the phenylalanine will get converted into tryptophan, right? So we know about 3 aromatic amino acid: tyrosine, tryptophan, phenylalanine. Interconversion is possible in between phenylalanine and tryptophan. If you add OH group, it will become tryptophan. If you minus OH group from the benzene ring, it will become phenylalanine. Okay. Now the most confusing topic about amino acid, that is ionizable nature of amino acid. So let's understood in a proper manner ionizable nature of amino acid. So what do you mean by ionizable nature? Let me clarify it. Suppose your son or daughter for your parents, but to your younger brother or younger sister, your elder brother or elder sister. So, the relation will vary. You are the same person, but the relation will be different for different person. So, that is the perspective you are observing you. If in the perspective of your parent, your mother and your father, you are just their son and daughter. For your younger brother and sister, you are elder brother or elder sister. For your elder brother or elder sister, you are the younger brother or younger sister. Similarly, if you keep amino acid into different kinds of solution, into different electric field, the nature of amino acid will change. So, that is why we are saying that sometime one particular amino acid will behave like positive ion or cation, sometime that same amino acid will behave like a negative ion or anion, sometime the amino acid will behave like it has one positive ion and one negative ion with it as a dipolar molecule. So, that is called as ionizable nature. I am again repeating it, sometime the amino acid will behave like positive ion, sometime the amino acid behave like negative ion, and sometime the amino acid behave like it has two polar end with it, one positive and one negative as dipolar molecule. So, that is called ionizable nature of an amino acid. Okay. Now, when an amino acid will behave like positive, when that same will behave like negative, and when the same will behave like a dipolar or Zwitter. Let us study that. So, if you are putting the amino acid inside an acidic solution, now we know that these acids are famous for donating H plus. If you go to the acid, then acid will say you, hey, I have many H plus ions with me, so please take H plus ion. So, acids are always donating H plus ions to you. So, if you keep amino acid into acidic solution, so acid will donate H plus ion. Now, who will eagerly take that H plus ion or acidic ion? Obviously, carboxylic group will not take, it is already acidic one. Now, who has hunger for acid? That basic part, NH2 ammonium group. So, it will take that H plus ion and it will become NH3 plus. Now, when it has become NH3 plus, the amino acid will behave like positive ion, and in our language we can call as the male amino acid. When the amino acid is behaving like positive ion, you can also call as male amino acid. Okay. Now, if you keep the amino acid into a neutral solution or isoelectric point, then what will happen? In neutral solution, the amino acid, the acidic group of amino acid will try to donate H plus ion to the neutral solution. So, here who is acid in amino acid? Carboxylic group is acid in amino acid. So, by donating H plus ion to this neutral solution, it will become COO minus. So, in neutral solution or isoelectric point, now if you observe this particular amino acid, this amino acid has one negative end CO minus and another positive end NH3 plus. So, in neutral solution or isoelectric point, one amino acid has one hand with positive ion, one hand with negative ion, negative group, and we call this amino acid as Zwitter ion. Now, Zwitter is basically a German word. Zwitter means bisexual. We are considering plus as male, minus as female. Now, if you transfer this particular amino acid into a basic solution, now what you can observe, now this basic solution will eagerly exhaust the extra H plus ion present into this amino acid. It will take the extra H plus ion, before it was NH3 plus. Now, the basic solution has taken extra H plus, now again NH3 plus has become NH2, Now the amino acid has only one end, that is COO minus. So you can call this amino acid as it's behaving as negative charge or a female ion. So that is called the ionizable nature of amino acid. Sometime it will behave like positive ion, sometime it will behave like negative ion, sometime it will behave like zwitterion. You can also call it dipolar because it has one positive pole, one negative pole. So that is the ionizable nature. In different solution, amino acid is behaving in different way. Okay. Now we will talk about protein. So we know that proteins are basically large giant polypeptide chains which are made up of multiple amino acids of different nature. Yes, so that's why it's written here, proteins are heteropolymer of amino acids. Proteins are heteropolymers of amino acid. If you want to construct a protein, you need to use multiple amino acids. We know about many amino acids, right? So if you are constructing a protein chain by taking only alanine amino acid, then it won't be forming anything. So to form a perfect protein chain or polypeptide chain, we call the protein chains as polypeptide chain, right? Remember it. Protein chains made up of multiple amino acids are called polypeptide chains. Here you can use alanine, valine, lysine, histidine, valine, tryptophan, alanine, phenylalanine, valine. So you can observe multiple amino acids are used to form this polypeptide chain. So now you can name it as a protein. So the bonds can be used, so bonds must be used to form, to connect these amino acids with each other, that is called peptide bond. We will understand what is peptide bond and how it is formed. So when you are connecting one amino acid with another one, the bond is formed, that is called peptide bond. Okay. So a basic structure of peptide chain is given here. So basically this is the first amino acid. From left hand side we have to start counting it and we have to end at right hand side. So the very left hand side, this will be our first amino acid. Then after that there will be one second amino acid. After that there will be one third amino acid and the chain will keep on elongating, so on. So if you are observing one polypeptide chain or protein chain into a linear manner, towards the left hand side you can observe there is a free amino group, there is a free amino group, and that is called N-terminal. And if you are observing the right hand side, there is a free carboxylic group, and that is called C-terminal. So where you are going to get N-terminal in a protein chain and in linear structure, in the left hand side you will get N-terminal. Why it's called N-terminal? Because there is a free amino group is present. Why right hand side is called as C-terminal? Because you will find a free carboxylic group here. Okay. Now let's understand how peptide bonds are formed. So basically this is the peptide bond. This is one peptide bond. This is another peptide bond. So let's understand how these peptide bonds between different amino acids are formed. So let's take one amino acid. Here I am putting the amino group, here hydrogen, here R group, here carboxylic group. So we will write the carboxylic group into this way, C double bond O and COH. We have broken down the generalized structure of the carboxylic group. Here I am putting the next amino acid, carbon, amino group I am writing into a broken structure, NH2, here H, here R group, and here normal carboxylic group. So this is the first amino acid, this is the second amino acid. We have to form peptide bond in between them. Now where this peptide bond formation will take place? The peptide bond formation will take place in between the carboxylic group of the first amino acid and the amino group of the next amino acid or the preceding amino acid. So peptide bonds, the direction will be from left hand side to right hand side, from left hand side to right hand side, and it will be in between carboxylic group and amino group. Remember it, peptide bond formation will take place in between the carboxylic group of one amino acid with the amino group of the preceding or the next amino acid. So here we have to do what? We have to do a dehydration. So easily you can see here probability of formation of water molecule, this OH and H, that will form water and that will get dehydrated. When that is getting dehydrated, a bond between here, a bond between carbon and this nitrogen molecule, I am focusing here, just observe here. When this part is dehydrated, there will be reaction between the hydroxide of carboxyl and hydrogen of amino group. The water will go out and there will be formation of this kind of structure. So this is the structure of peptide bond. So this is how peptide bond is formed in between two amino acids, and do not forget to remember about the N terminal and the C terminal. Okay. Now sometime you will be asked, define what is dipeptide, define what is tripeptide, define what is pentapeptide. So the very first diagram of amino acid, basically that is a tripeptide because we have used 3 amino acids here. So for a dipeptide there will be 2 amino acids, for a tetrapeptide there will be 4 amino acids. So it's a what kind of peptide? It's a tripeptide. So how many peptide bonds are present here? 2 peptide bonds are present here. 2 peptide bonds are present here. For a tripeptide there will be formation of 2 peptide bond. For a dipeptide there you need 2 amino acid and 1 peptide bond. For tripeptide you need 3 amino acid and 2 peptide bond. For pentapeptide you need 5 amino acid and 4 peptide bond. Do not get confused by the word peptide. Here peptide means the number of amino acids, here peptide does not mean the number of peptide bond. Okay. Now we have to study about protein in detail. Now you know that proteins have different structures. Proteins have primary structure, secondary structure, tertiary structure and quaternary structure. So when the crude protein is formed inside the cell with the help of ribosomes are called protein factory, right? They get consecutive modification with the help of Golgi complex, right? And primary structure of protein will be modified into secondary, then tertiary, then quaternary structures. The site of protein synthesis, we know there are 2 sites inside the cell, ribosome and RER, rough endoplasmic reticulum, because the endoplasmic reticulum surface has the ribosomes only. Okay, we will be only talking about 20 amino acid. Why so? Because they are very much needed for us for protein synthesis. So they are proteinogenic amino acids. Okay. Now further we can divide these 20 amino acids into 3 groups: non-essential amino acids, essential amino acids and semi-essential amino acids. Sometimes it is divided only into 2 groups, non-essential and essential. Now what is the difference in between them? When you are talking about non-essential amino acids, so non-essential amino acids means our body can produce them, inside our body, right? Inside our body is produced, or by the process of biosynthesis those amino acids can be produced. So we are calling them non-essential amino acids. Now which amino acids are called then essential ones? We cannot, or our body cannot produce them by the process of biosynthesis, so we need to take them through dietary source as dietary proteins. There is no other way, right? And few amino acids are called semi-essential amino acids. Only infants needs those amino acids, but when you are growing old, you no more you are needing them. So there are total 10 non-essential amino acids, 8 essential amino acids and 2 semi-essential amino acids. So let us talk about the examples of non-essential, essentials and semi-essentials. Okay. So when you are talking about essential amino acids, a great trick is here. It will help you to memorize their name, though it is not so much important to understand, because NCERT will not be saying about, or in NTA you are not asked about give some examples of essential or non-essential. Just for the sake of information we will observe that, we will study this. So the trick to remember the names of essential amino acid, that is TV till 8 p.m. So how many essential amino acids? You know, there are 8 essential amino acids. So 8 p.m. till 8 p.m. you have to watch TV till 8 p.m. Now let us understand what do you mean by TV. T for threonine, V for valine, till tryptophan, isoleucine, lysine and leucine, till. Then PM, phenylalanine and methionine, phenylalanine and methionine. Semi-essential, there are only 2, arginine and histidine, and rest all will be non-essential amino acids. And sometimes it is asked that give example of sulfur containing amino acid. So there are 2 examples are there, sulfur containing amino acids, cysteine and methionine. When you talk about cysteine, this is the structure of cysteine. With the R group you will get thiol group, CH2SH. Okay. Now let me clarify the difference between the essential and non-essential amino acid in a detailed manner. When we were talking about the aromatic amino acids, we got to know about this phenylalanine and tryptophan. So remember these two names, phenylalanine and tryptophan. Now the example of essential amino acid, you got the name of phenylalanine, because you need to take this phenylalanine through your dietary source, right? So once you got phenylalanine into your body, what you have to do? You have to just add extra hydroxide group with it and you will get the amino acid tryptophan, phenylalanine and tyrosine. Phenylalanine to tyrosine conversion will be there. So let's move forward. So phenylalanine, that's the essential one, and tyrosine, that's a non-essential one. You need just one enzyme, phenylalanine hydroxylase, phenylalanine hydroxylase, and there will be transfer of one hydroxide with phenylalanine structure with that benzene ring, and you will get the tyrosine. I think little bit correction is needed into that slide. I told just one wrong thing here. If you add hydroxyl group here, you can convert phenylalanine to tyrosine, not to tryptophan. Okay, so we are done with this. Now some informative things about proteins. So most abundant protein, that is one of the important enzyme, ribulose bisphosphate carboxylase oxygenase. Ribulose bisphosphate carboxylase oxygenase, most abundant protein in animal. Now when you are asked most abundant protein in animal, the answer will be collagen. The answer will be collagen, which is a very important fiber or very important protein of our intercellular substance or intercellular ground substance or intercellular matrix formation. Okay. So please try to understand the difference between most abundant protein in this biosphere and the most abundant protein only in animal kingdom. When you are asked most abundant protein in our body in animal kingdom, the answer will be collagen. Most abundant protein in this living world, in this biosphere, the answer will be rubisco, right? You need this rubisco for the process of photosynthesis. Okay. Now one more important statement, very important. Most enzymes are proteins except ribozyme. So if you get a statement all enzymes are protein, that's gonna be a wrong, completely wrong statement. Not all enzymes are proteins. Most of the enzymes are protein except ribozymes, because these are basically RNA. These are ribonucleic acids. Ribozymes are ribonucleic acids. Okay. Now function of few important proteins of our body. Collagen is the component of ground substance. Even collagen protein will make collagen fiber. Collagen can give rise different types of fiber. Collagen fiber, modified collagen can give rise reticular fiber. Remember it. And these collagen fibers you need for the formation of bone. You will find them into matrix of bone, inside the matrix of cartilage, right? Trypsin, protein digesting enzyme, one of the protease, protein digesting enzyme. Trypsin, you study in the chapter of digestion. Insulin is a hypoglycemic hormone. Insulin, that reduces blood glucose. That's why we are calling it as hypoglycemic hormone. Insulin is secreted by beta cells of pancreas. Pancreas have some special endocrine tissue called islets or islets of Langerhans, not islets, islets of Langerhans, that is alpha cell, beta cell, delta cell. So these beta cells are responsible for the production of insulin. Antibodies, they fight against infection, infections. We also call them gamma globulins or immunoglobulins. Their common signal is Ig. Now, when you talk about Ig, there are different types of bodies are present into our body. We call them immunoglobulins. Remember this term, GUMD, IgG, IgA, IgM, IgD, IgE, GUMD. So this is also the sequence of relative abundances of these antibodies, right? The most abundant antibody IgG, then second most abundant IgA, third most abundant antibody IgM, fourth most abundant antibody is IgD, and the rarest antibody which is also responsible for allergic reaction, that is IgE. Okay. Now the receptors present on the cell surface, they are also mostly made up of proteins or glycoproteins. So receptors, they can be sensing receptors, taste receptor, smell receptors, receptors on our taste buds, smell receptors on our olfactory epithelium of nose. Okay, GLUT4 is connected to insulin. So I said that insulin is a hypoglycemic hormone, right? So if this is the hepatocyte or liver cell, liver cells are called hepatocytes. Now glucose is present into our bloodstream. Now when influenced by insulin, basically insulin will stimulate the hepatocyte cells or our muscle cells to produce some glucose transporter proteins on their cell membrane, and we call them GLUT. So GLUT means glucose transporter. Glucose transporter, shortcut form is GLUT, glucose transporter type 4. So basically what is the function of this glucose transporter type 4? With the help of this GLUT4, glucose is transported from blood inside the cell. So glucose transporter can work in different direction. Few glucose transporter will transport glucose from cell to blood, and few glucose transporter will transport glucose from blood to cell. Cells means here they will transport glucose inside the hepatocytes or liver cells. Hepatocytes, the cells of liver, inside the myocytes or muscle cells. Okay. Now let us move forward to the next topic. Now we will talk about structure of protein, starting from the primary structure to secondary and tertiary structure. Okay. So just it's a very simple thing. So when you talk about primary structure of protein, that we call 1 degree structure, that's basically straight chain or linear chain protein. When you form skink or coiling or a complexity into it, that will become the secondary structure of protein. the protein will be called as secondary structure, and after that when you are performing further complexity generation, we call that polypeptide chain as tertiary or 3-degree structure, right. So when you are playing with one single polypeptide chain, the most unstable, non-functional functional or crude protein polypeptide chain is called tertiary protein. When it got a little bit more stability by forming few more bonds, we call it secondary structure of protein, and the functional, most of the proteins are functional in tertiary structure. Most of the functional proteins or enzymes of our body, they are tertiary protein. They got further complexity by forming many more bonds, right. Now what after tertiary? We call it quaternary or 4-degree protein. Now for this you need more than one, for this you need more than one tertiary proteins. So when more than one tertiary proteins are linked, more than one tertiary proteins are linked, that will form a further stronger and complex structure, that is called quaternary structure. So that was the very basic thing about the structure of proteins. Now let us study in detail. So first we will talk about the primary structure of protein. So primary structures, basically they are linear chains, linear chain made up of multiple amino acids. So this is the linear chain. Now what is the significance of primary structure of protein? If you observe this linear chain, you know the left hand side or towards the end terminal, this is the first amino acid, and towards the right hand side or the C terminal, this is the last amino acid. So that is called positional information of amino acids can be obtained from the primary structure of protein, and the bond is used here to connect these amino acid molecules, that is simply peptide bond. Got it? Now let us move to the complexity from primary to secondary structure. Now to form the secondary structure of protein, you need few more bonds. Along with peptide bonds, you have to use hydrogen bonds. So one more bond is added. Already we have the peptide bond in primary structure. Now you have to utilize hydrogen bond here. Basically hydrogen bond is not a typical chemical bond, it is a kind of attraction force. Okay. So when you are forming this vertical hydrogen bond, when the vertical hydrogen bonds are produced in between the special amino acids present into these polypeptide chains, then that will form a special structure called alpha helix. So in alpha helix, almost they are right-handed helix. The proteins, the alpha helical proteins in nature, they are right-handed helix. So this is the right-handed turn, and the hydrogen bonds are present here. They are vertical hydrogen bonds. Okay. Now another structure you can observe here. This is called beta pleated sheet. Now what about this beta pleated sheet? When horizontal hydrogen bonds are produced in between polypeptide chains or within polypeptide chain, that will form a sheet kind of structure, then that is called beta pleated sheet. So here we will find horizontal hydrogen bond, horizontal hydrogen bonds. So that is the difference between alpha helix and beta pleated sheet. Alpha helix is a right-handed helix, beta pleated sheet is a sheet. For alpha helix you need only one polypeptide chain. For beta pleated sheets, sometime more than one polypeptide chains can be used to form these horizontal hydrogen bonds, and even with one polypeptide chain this beta pleated sheet can be formed. The difference of hydrogen bond: for beta pleated sheet horizontal hydrogen bond, for alpha helix vertical hydrogen bonds. Vertical hydrogen bonds, horizontal hydrogen bonds. When you are standing, that is the vertical position. When you are sleeping, that is the horizontal position. Okay. Now let's come to more complexity, that is tertiary structure of protein. Tertiary structure of protein. So basically that will resemble you like a woolen ball like structure. Tertiary protein, further complexity had appeared in between the alpha helix and beta pleated sheet, and more bonds formation will be there. Up to secondary structure we only got peptide bond and hydrogen bond, right? Now it's time to form few more new bonds. Here there will be formation of sulfur, that is disulfide bond. Few ionic bonds will be formed, few hydrophobic bonds will be there. Okay, out of these bonds the strongest bonds are peptide bond and disulfide bonds, because they are covalent bond. Remember, peptide bond is the strongest because peptide bond as well as disulfide bond, they are covalent bonds, rest are attraction forces, right? Hydrophobic bonds, another kind of attraction force. Hydrogen bond, a different kind of attraction force. Even van der Waals force will take part by forming sometime when you are forming tertiary structure of protein. Okay. Now when we are talking about one single polypeptide chain, this is the most stable, active and functional form of polypeptide chain. It's very much stable, that's why most of the functional proteins like enzymes are present in tertiary format, right? But when you talk about more than one polypeptide chain, we have to discuss about the quaternary structure of protein. So when more than one tertiary polypeptide chains are bound together with the help of bonds, obviously they can form disulfide bond. Possibilities are there will be simple hydrogen bonds. Apart from the, obviously the previous bonds are present, the bonds which are present in tertiary, all of them are present into quaternary structure. Now to bind different tertiary proteins you need to produce few more bonds, like right? So that's why you need more disulfide bonds or more hydrophobic bonds or more ionic bonds to configure the quaternary protein. So here one structure of quaternary protein by three tertiary structures are exhibited. So as because maximum number of bonds are present, so this is the most stable structure, remember. So when you are talking about the structure of protein, which is the most stable structure of protein, obviously quaternary structure. But if the question is twisted, which is the most stable structure in case of one polypeptide chain of protein, then the answer will be tertiary structure. Obviously minorly you have to read out the sentence, right? Some examples are given here, insulin and hemoglobin. So when you talk about insulin, it has a B chain, B polypeptide chain and A polypeptide chain. When you talk about hemoglobin, that is a tetramer, it has two alpha chain and two beta chain, right? So that is the normal structure of hemoglobin. So insulin is a dimer, hemoglobin is a tetramer of quaternary structure of protein. Okay, so that's how we are done with the structure of protein. Now we will talk about lipids. Let's start with lipid. So we have to talk about three forms of lipid: simple lipid, conjugated lipid, and derived lipid or lipid derivatives. So first talk about simple lipids. Which lipids are considered as simple lipids? One very wrong conception is there in us that lipid, first thing, lipid is not a monomer. Lipid is not a polymer. Remember this. Lipids are obviously biomicromolecule, and lipids can be fatty acid, lipids can be glycerol. Fatty acids are lipids, glycerol is also lipid, fat is also lipid, oil is also lipid, wax is also lipid. So in generalized manner we say that when we break down lipid with lipase, we will get the monomers called fatty acids and glycerol. That is a wrong statement, mind it, because fatty acid is also a lipid, glycerol is also a lipid. So let us understand what about different types of lipids. So fatty acids, glycerol, fat, oil and wax, all of them are considered as natural lipids or simple lipids. Okay. Now what is conjugated lipids? A complex lipid with one lipid part and another non-lipid part, that is called conjugated lipids, right? So very important point to understand, one lipid plus another non-lipid, that will form conjugated lipid. One of the example is phospholipid. Phospholipid, lipid plus phosphate group or phosphoric acid. Lipid plus phosphate group or phosphoric acid will form phospholipid. Glycolipid, lipid plus carbohydrate, that is called glycolipid. And lipoprotein, lipid plus protein, that is called lipoprotein. So that's why we are calling them conjugated or complex lipid. Now derived lipids are basically lipid derivatives. They have lipid kind of function, okay, but they don't have any sort of fatty acids molecule or glycerol molecule inside them. So basically the biosynthetic process of our body is very much complex for us. So few of those biosynthetic processes will start the reaction, like chain with a lipid molecule, and it will keep on converting, keep on adding and deleting groups into it, and ultimately that will give rise molecule which has function like lipids but no structural similarities with fatty acids or glycerol. So that's why we call them derivatives of lipid or lipid derivative. So one of the example is given here, cholesterol. There can be sterols or sterons. So few examples of cholesterols are here, like not examples. Cholesterol is derived lipid present into our body, and cholesterols help us to form steroid hormones. Like if you study the process of biosynthesis of estrogen, study the process of biosynthesis of testosterone or progesterone, cortisol, you will find that it starts with cholesterol, a steroid moiety. Okay, cholesterols are needed for production of vitamin D. So precursor molecule of vitamin D, cholesterol, as well as cholesterol also act as precursor molecule for bile salts. Okay, what you have to know only about cholesterol in your NCERT, the structure of cholesterol is given here with multiple honeycomb like structure. You will find that a multiple ring structure. Okay, a beautiful multiple ring structure. That is the structure of cholesterol. Obviously we will study about that. So we start with fatty acid. We will start with fatty acids. The generalized formula of fatty acids, is RCOOH. Here R is the alkyl group and COH is the carboxyl group. You already know that R is the alkyl group and COH is the carboxyl group. Okay. Now the carboxyl group will be constant for all the fatty acids. What is variable here? Again, here R group is variable group. You have to change R group to form different fatty acids. Like common formula of R is CnH2n plus 1. So sometime the R group will be CH3, sometime the R group will be C2H5, sometime the R group will be C3H7, sometime the R group will be C4H9 and so on, right. So what you have to do, you have to keep the carboxylic group constant, you have to keep changing the R group to get different fatty acids. So it's written here, R group of fatty acids can be formed. Now when you are forming R group, when you are taking R group, you have a limit, that is the smallest R group can be of 1 carbon and the longest R group can be of 19 carbon, right. Now the example is here, when you are forming acetic acid. Acetic acid is a 2 carbon fatty acid. Total carbon in acetic acid, that is 2 carbon. So 1 carbon you got from this carboxylic group. Now what about the another carbon? That is the smallest R group, that is methyl group CH3. Here you got another carbon, here you got the second carbon. So every fatty acid molecule have a constant carbon, 1 carbon present into the carboxylic group, and the remaining carbons, they are present inside the R group. Let us take example of propionic acid. Let us take an example of propionic acid. There is a 3 carbon fatty acid. When you are talking about propionic acid, there will be 2 carbon of R plus 1 carbon of carboxylic group, right. So the R group used here, that is ethyl group C2H5. You can also write it like this, CH3 CH2 COOH. In this manner also you can write the similar structural formula C2H5COOH. So that is the structure of propionic acid. Let us move forward. Example is given in NCERT, that is palmitic acid. Okay, very important. Multiple time questions has been asked from this. So when you are talking about palmitic acid, so basically that is a 16 carbon fatty acid and the R group has 15 carbon. The formula of R group of palmitic acid is C15H31 and obviously that has the constant carboxylic group. So in case of palmitic acid, how many carbon from R group? 15 carbon from R group and 1 carbon from this carboxylic group, right. Okay, the complex one, the most complex fatty acid is considered when we are talking about arachidonic acid, that is 20 carbon fatty acid. There is another 20 carbon fatty acid example you are going to get, that is arachidic acid. There are 2 similar named acid. One is arachidonic acid, that is also 20 carbon fatty acid. Another is arachidic acid, that also has 20 carbon, but the complex one in structural complexity will observe in arachidonic acid. Here R group has that 19 carbon and 1 carbon into the carboxylic group. Okay. So in NCERT, 2 examples are very much important. One is of palmitic acid and another is of arachidonic acid. Okay. Now let us classify the fatty acids. Fatty acids are classified into 2 groups, saturated fatty acids and unsaturated fatty acids. Okay. Now what about this saturation and unsaturation? Here you have to talk about the carbon, the type of carbon bonds present into them. So please look at the structure given under the saturated one. So here you are going to get the carbon-carbon bonds are always single bonds. So if there is only single bonds, in between carbons, then that fatty acid is say it to be a saturated one. Okay. Now these saturated fatty acids are solid in room temperature. Now that's give them a medical harmfulness. Medical condition can be arised like the diseases CAD, coronary artery disease. You know, you have already studied in the chapter of body fluids and circulation. You have studied about the atherosclerosis, the deposition of bad cholesterol or bad fats into our blood vessels. Okay, that is due to the presence of too much saturated fat, right? So you need to avoid consumption of too much saturated fat. So what it can cause? If you observe your blood vessel, saturated fat, as because they are solidified, they can form these fatty plugs or solid mass of fat, a lipid inside the blood vessel, and that will cause disturbance in the pathway of blood flow and sudden hypertension, blockage of coronary arteries, and a disease called CAD. So please be aware that avoid saturated fatty acids, or these are also called as trans fats. These are present in cheesy material, in animal fat, or in fast food materials. Okay, they are rich in palmitic acid and stearic acid. That's why whenever you are eating something from a fast food shop, or you are buying the cooking oil, you have to check what percentage of impurities are present. Basically they mix. Suppose you are buying mustard oil, if you are buying rice bran oil, if you are buying extra virgin olive oil. Now if some unscrupulous businessman want to gain more profit, inside that olive oil he or she will mix palmitic acid or palm oil as impurities. Now if you take out the palm oil, if you buy palm oil, it's very cheap in market, and if you keep it in room temperature, specifically in winter season, that will get solidified in room temperature. But have you ever observed that mustard oil is getting solidified in room temperature, olive oil is getting solidified in room temperature? Never, because that has very less quantity of saturated fats. So avoid the oils or fats which are rich with saturated fatty acids. Examples of palmitic acids and stearic acid. Okay. Now let's come to the next one, that is unsaturated fats. These are quite healthy for our body. Again, these unsaturated fats are of two types, PUFA and MUFA. Polyunsaturated fatty acids and monounsaturated fatty acid. PUFA for polyunsaturated fatty acids, MUFA for monounsaturated fatty acids. Often it's said that PUFA, polyunsaturated fatty acids, are better for us. Okay. Now, what is the reason behind this unsaturation? If you observe the structure, you will find single bonds are there in between the carbon atoms, but also you will find double bonds. So, if one or more than one carbon-carbon double bonds are present, then that fatty acid can be called as unsaturated fatty acid. If one double bond is present, that is called MUFA. If more than one double bond is present, that is called PUFA, polyunsaturated and monounsaturated. Okay. Now few of the examples are given here: arachidonic acid, oleic acid, linolenic acid, linoleic acid, and you will find them, most of them are liquid in room temperature. That's why there is less risk of getting solidified or getting or forming the fat plaque or cholesterol plaque inside your blood vessels. Now what is the reason behind that? The saturated fats, saturated fatty acids, they will form solid crystals, or they will solidify in room temperature, but unsaturated fat, they won't get solidified because the production of these kinks or turns wherever you will find these double bonds. So it's as easy, as simple as that. If this is a saturated fatty acid molecule, and if you keep multiple saturated fatty acid molecule together within a range of temperature, they will adjust them with each other to form a proper crystallized structure, and that will form a solid mass. But as because the unsaturated fatty acids, they have skink wherever they are forming these double bonds, it will be very tough for them for sit together, to adjust together to form a solid structure. That's why they remain liquid in room temperature. So we are done with fatty acids. Now we'll talk about glycerol. Okay. So when you are telling fatty acids, there are multiple fatty acids are there, but there is only one glycerol. So the common name, the chemical name of glycerol is trihydroxypropane, and the formula is C3H8O3. So here you can see there are 3 carbons and there are 3 hydroxyl group is there. So that's why you are calling them trihydroxypropane. Propane, 3 carbon. Okay. Now we are done with glycerol. Now we'll talk about fat and oil. So basically fat and oil, technically they are similar, technically they are called glyceride, and the natural fat and oil, they are present in the form of triglyceride. So what is the difference between them if they are similar thing technically? So the difference is that fat, they have high melting point, but oil, they have low melting point. So that is the difference between fat and oil. Fats in room temperature, when you observe them, they will look like a solid mass, but oil in normal temperature, they appear liquid, right? Now how you can produce fat or oil? By utilizing glycerol and fatty acid. So you have to perform a esterification reaction between fatty acids and glycerol, and we will obtain fat or oil. That's why we are calling them esterified fatty acids and glycerol. So you need to perform another reaction between glycerol and fatty acids. Now if you are asked that give some examples of common fat, then you can obviously tell butter, ghee and other animal fats. Oils, example of oils, mustard oil obtained from the extract of mustard seeds and gingerly oils. Okay. Now let us understand how esterification between fatty acid and glycerol is done. So the molecular structure you can observe here, that is the structure of triglyceride. That is the structure of triglyceride, because there is only one molecule of glycerol and all the hydroxide groups are occupied with fatty acids. So this is fatty acid 1, this is fatty acid 2, this is fatty acid 3. So when 3 different fatty acids will form ester bond with glycerol molecule with the hydroxide group, then we will call it triglyceride. Now let us understand how this esterification reaction is happening. So this is glycerol CH3OH, sorry, CH2OH, CHOH and CH2OH. Now if you want to form a monoglyceride, you want esterification of one fatty acid with this glycerol molecule. So you have to take a fatty acid molecule. So let's arrange fatty acid molecule like this. This is the fatty acid molecule. You have to perform what? You have to perform esterification. For esterification, what you need? You need another dehydration here. So perfectly you can observe that dehydration is possible in between the hydroxide group and the carboxylic group of fatty acid, right? When water will go out, there will be formation of bond. Now this is perfectly esterified. Now this particular composition can be called as monoglyceride, right? Now if you perform similar thing with this hydroxide group with another fatty acid, then that will be called diglyceride, and all the three hydroxide groups if occupied with three different fatty acid molecules, then we call that triglyceride, and this is the form the natural fats or oils are present into our body as triglyceride form. Okay. Now let's talk about conjugated lipids. Already we have spoken about them. Conjugated lipid means conjugation of lipid molecule with a non-lipid molecule. That is called conjugated lipid. So the example is given in NCERT, that is a phospholipid. Now where in our body you can get phospholipids? We will find the phospholipid into our cell membrane, and that is called lecithin. So when you are studying your cell, you know about the phospholipid bilayer present into your cell membrane, right? So this is one of the phospholipid molecule. And this is the head of phospholipid, this is the neck of phospholipid molecule, and this is the tail of phospholipid molecule. Now what is the nature of head? What is the nature of tail? So we know that tail is hydrophobic in nature, but the head is hydrophilic in nature. So let's discuss why the head of fatty acid is hydrophilic but tail of fatty acid is tail of phospholipid is hydrophobic. Head of phospholipid is hydrophilic, tail of phospholipid is hydrophobic. Okay, so it's the structure is kind of similar to triglyceride, but in the position of third hydroxide group, you have to replace it with phosphoric acid of phosphate group, and after phosphate you have to add another group that is called choline group. Now your phospholipid molecule is produced. See, this part and this part is similar to glyceride molecule. This is the glycerol, part, glycerol moiety. First fatty acid esterified, second fatty acid esterified. Now the third position, what you have done here, you have created a bond between phosphoric acid and the hydroxide group. So this is the addition of phosphate group. So wherever in the molecular structure you will get this structure, appearance of phosphate group, then blindly you can say this is the phospholipid molecule or lecithin molecule. Okay. Now what about this particular structure? This particular structure is called choline. This particular structure is called choline, that is making the head, of phospholipid molecule, and this choline has this choline group has a hydrophilic nature. Now, as because the tail is made by the glycerol and fatty acid, then obviously these glycerol and fatty acids are hydrophobic in nature, and this phosphate group that is responsible for the formation of neck region of this phospholipid molecule. Okay. Now let's move forward. Another conjugated lipid we have to talk about, that is sphingolipid. There is a conjugation of lipid molecule with sphingolene. Okay, where you can get this sphingolipid into our neural tissue, and I think you all know about this term myelin sheath when you talk about myelinated neurons. Yes, this white colored sphingolipid myelin is responsible for the process of myelin sheath formation upon the axon. And this myelin sheath, you know very well, this myelin sheath, they are secreted by this myelin are produced by Schwann cells. So these are Schwann cells, and these Schwann cells will produce layer wise myelin sheath upon axolemma, and this is a structure of neuron, diagram of neuron, and this kind of neurons are called myelinated neuron. And you know this myelinated neuron are very fast in the process of nerve impulse propagation. Okay. So you are done with sphingolipid. Now let's talk about the cholesterol molecule. Yes, so that is one of the fine example of derived lipid or lipid derivative. Already we have discussed about. Now just you have to practice again and again the diagram of cholesterol. How many ring structures are present into this? 4 rings are present, and 3 are hexagon, 1 is pentagon, and the 5th one is the incomplete pentagon, incomplete hexagon, one arm is absent. So whenever you observe this particular structure, this particular moiety is called steroid nucleus, and this is the structure of cholesterol, which is a precursor molecule of many steroid hormones like estrogen, progesterone, cortisol. This is the precursor molecule of vitamin D. This is the precursor molecule of bile salts. Okay, so we are done with lipids. Now we'll move forwards to the energy molecules, that is carbohydrates, carbs. Yes, now how you can define carbohydrates? So basically they are polyhydroxyaldehydes or ketone, or their polymers. So carbohydrates, when you talk about monosaccharides, they are polyhydroxy aldehydes or ketones, or they are polymers of polyhydroxyaldehydes and ketone. Now why I am telling they are polyhydroxyaldehyde and ketone? Let's understand with the structure. So this is aldose sugar or glucose, and this is a ketose sugar or fructose. Now here you can observe very easily, very clearly, that addition of one aldehyde group with glucose and addition of one ketone group with fructose. So as because the glucose has this aldehyde group, we call it aldose. Obviously it's a hexose sugar with chemical formula C6H12O6. Again, if we are talking about fructose, it is also another hexose sugar, similar formula with C6H12O6, right? But the different structure. Here the functional group is ketone group, so that's why glucose is aldose but fructose is ketose. And you can find that multiple hydroxide groups are present here, so you can obviously call them polyhydroxy aldehydes or ketones. Okay, now you can polymerize this glucose or fructose. So like glucose is polymerized to form cellulose, starch, fructose is polymerized to form inulin. So that's why the definition is like this. Okay, so there are 4 types of monosaccharides we need to talk about. 4 types of carbohydrates we need to talk about. They are monosaccharides, disaccharides, oligosaccharides. Sometimes we consider disaccharides under oligosaccharides, and obviously the polymers, they are called polysaccharides. They are the giant molecules. So have to emphasize specifically on polysaccharide molecules. Okay. Now, which carbohydrates are called monosaccharides? Okay. So basically, again we need to talk about polymer and monomer concept. Polysaccharides, oligosaccharides or disaccharides, these are polymers, and if you hydrolyze, if you break the units of polymer, you will get monomer. So upon hydrolysis of polymer, we will get the beads, the units like structure, they are called monomer. You have to do hydrolysis. Okay. But when I am talking about monosaccharides, they are the building blocks of disaccharides, oligosaccharides and polysaccharides. So further hydrolysis of monosaccharides not possible. You cannot hydrolyze them. Hydrolysis is not possible. You cannot further break them down to obtain their unit material. So that's why the smallest among carbohydrates or the units of polysaccharides, oligosaccharides or disaccharides can be called as monosaccharides. Mono means 1. Mono means 1. So that's why it's written here, hydrolysis is not possible, only made up of 1 unit, 1 single unit, right? Examples, there are multiple examples are there: glucose, fructose, galactose, ribose, deoxyribose, mannose, and all of them are reducing sugars. So monosaccharides are reducing sugar, and generally they will get dissolved in water. Now disaccharides, you need 2 units of monosaccharides. Okay, when you add 2 units of monosaccharide by the bond called glycosidic bond, so if there is a glycosidic linkage between 2 monosaccharides, that will form disaccharides. So di means 2. Okay, examples: sucrose. What is another name of sucrose? Table sugar. Maltose, lactose, another name of lactose, the sugar present in milk or milk sugar. So now oligosaccharide, oligo means few. So for oligosaccharides, you will get 3 to 10 units of monosaccharides. By multiple glycosidic bonds, and in nature you will find these oligosaccharides is a part of cell membrane. Like when you study the structure of cell membrane, you will find with cell membrane structure, with phospholipids and with proteins, few branched structures are attached. So these are called oligosaccharides. One of their example is raffinose. Raffinose, stachyose, some of the examples of oligosaccharides. Now let's talk about polysaccharides. So poly means multiple. When there is more than 10 or many monosaccharides are combined together to form a carbohydrate molecule, a long chain of carbohydrate molecule, by utilizing glycosidic linkage, we call them polysaccharides. So more than 10 monosaccharides are needed. More than 10 monosaccharides are needed. Yes. Examples: starch, the storage molecule of glucose is a polymer of glucose. Cellulose, structural polymer of plant. Glycogen, It's also called as animal starch, a storage carbohydrate, chitin, structural carbohydrate in some animals like arthropods. You will find chitin in the cell wall of fungi, and a polymer of fructose, there is inulin, you will find in the root tubers of dahlia and artichoke plants. Okay. So let us understand the molecular structure of some monosaccharides, because though it is not given in our syllabus, but we have to know how to construct the long chain molecule of polysaccharides. So when you are constructing starch, when you are constructing cellulose or glycogen or inulin or chitin, you need a basic knowledge of their monomers, right. So we will study the structure of glucose, structure of fructose, structure of galactose first, and then structure of N-acetyl glucosamine. So let us see. So all of them are basically hexose sugar. All of them are hexose sugar. They have similar formula. Either you can call tell about glucose or fructose or galactose. Glucose, fructose, galactose, they are isomeric molecule. They have similar formula of C6H12O6. For all of them, the formula will be C6H12O6, but because they are structural isomer, the structure will vary from each other. Okay. Now, so this is the hexagonal structure of glucose. Now let us understand how glucose molecules are formed. The ring structure of glucose. So that is a hexagon. This is the ring of glucose. And glucose is 6 carbon molecule. So this is the first carbon, this is the second carbon, this is the third carbon, this is the fourth carbon, this is the fifth carbon, and this is the sixth carbon. That is why we call it a hexose sugar. Means 6 carbon is present. Okay. Now if you observe the ring, how many carbons are present inside this ring? 1, 2, 3, 4, 5 carbons. 5 carbons inside the ring. So this kind of rings are called, here it is written pyranose ring. So 5 carbon present inside the ring, they are called as pyranose ring. You have to put the remaining positions with hydroxide and hydrogen. Now you got the structure of glucose. So this is the ring structure of glucose. Now if you modify in third carbon, you will get galactose. If you modify in second carbon, you will get mannose, right. Okay, so this is the structure or pyranose structure, ring structure of glucose. Now let us talk about fructose. This is the first carbon, this is the first carbon, this is the second carbon, this is the third carbon, this is the fourth carbon, this is the fifth carbon, and this is the sixth carbon. So this is the structure of fructose molecule, and this is also hexose sugar, because total there are 6 carbons. So this is also hexose sugar, right. Fructose is also a hexose sugar, and about the ring. About the ring. How many carbons inside the ring? 1, 2, 3, 4. Though it's a hexose sugar, but the ring structure, if you observe only, 4 carbons are present inside the ring. So that is the furanose ring. 4 carbons inside the ring furanose, 5 carbon inside the ring pyranose. So the heterocyclic structure of glucose, that is a pyranose ring. Heterocyclic structure of fructose, that is called furanose ring, right? Why I am calling it cyclic ring? Because inside the ring, apart from carbon, it also has the oxygen oxygen atom. That's why it's a heterocyclic. If the ring is made up of only carbon, that is called homocyclic ring. But this ring has oxygen apart from the carbon, so it's a heterocyclic ring. So I think you understood what is furanose and what is pyranose, right? So this is the structure of glucose, this is the structure of fructose, we have already discussed. Now this is the structure of galactose. I said there will be modification in which carbon? Third carbon. Now if you observe the modification in fourth carbon, the difference between glucose and galactose. Now you have to observe the structure of the orientation of OH group. So for glucose in fourth carbon, the OH is present below the midline, and for galactose the OH group is present above the midline. So that's why there is change of orientation of H and OH group in fourth carbon, right? So that will modify glucose into galactose. Next, NAG. Now what is NAG? N-acetyl glucosamine. So this is called amino sugar. This is a modification of glucose molecule. So rest everything is similar. The second carbon, you have to add one amino group and acetyl group. So this is the amino group and this is the acetyl group. So that's why it's named as N-acetyl. This is overall the skeletal component is glucose. So in second carbon you have added amino group and acetyl group. So its acetyl group is added with the nitrogen. So it's N-acetyl and it's a glucose, and this is the amino group. So it's glucose amine and it's called as amino sugar. Okay. Now we'll talk about two pentose sugar. One is ribose and another is deoxyribose, which will use the structural component as structural component of RNA ribose, and in DNA we will use deoxyribose. Now they are called as pentose sugar because they have 5 carbon present inside them. So that's why they are called pentose sugar. So this is ribose, this is deoxyribose. What is difference in between them? If you observe the second carbon, for ribose there is a hydroxide group, and if you observe the second carbon position of deoxyribose sugar, there is only H group. So one oxygen is deleted from deoxyribose from the second carbon position of ring, that's why you are calling it, the name is 2-deoxyribose sugar. So only the difference is the structural formula. If you observe C5H10O5, the formula of ribose sugar. Now if you delete one oxygen, then it will become C5H10O4, the formula of deoxyribose sugar, and all of them are examples of monomer. Okay. Now let's go dive inside the glucose molecule. So whatever we have gathered the knowledge that we know that glucose, hexose sugar, as because glucose molecules are made up of 6 carbon, the ring structure has the 5 carbon inside, so they are called pyranose ring. Now this glucose, the ring structure of glucose are also optically active structure. You know what is optical isomerism? One is a right-handed structure and another is a left-handed structure. So when we will pass polarized light within the molecule and the rotation of the molecule will take place. So one will rotate into the left side, another will rotate to the right side. So that is called optical isomers, and we call them D form and L form. And you have to understand one more important thing: the glucose which is present in nature, they are mostly present as alpha glucose. So whatever molecules we are going to talk about in our study, they are the molecules, they are not alpha glucose, they are present in D form, dextrorotatory form. Yes, those are natural glucose molecules, they are D forms. Even in this chapter biomolecules, natural carbohydrates, they are present in D forms, right. Okay. Now we have to differentiate between this alpha D glucose and beta D glucose. Now what makes them alpha form and beta form, right? So all the glucose cyclic structures we are going to study, they are D glucose, but we have to differentiate between which is alpha glucose and which is beta glucose. Now where you have to focus? You have to focus on first carbon. You have to focus on first carbon. Now let us make a boundary. Let us draw a midline of this ring structure. So this is the midline, the equator of this ring structure. Now you have to observe the position of hydroxide group. It's above the midline or below the midline. If it's below the midline, then the structure is called alpha glucose. If hydroxide group is above the midline, then the structure is called beta glucose. Is it clear? So we have to focus on hydroxide group attached with first carbon. So if the position is above the midline, beta, below the midline, alpha. Which hydroxide? First carbon hydroxide. Okay. Now let us talk about disaccharides. So for all the disaccharides, the formula is similar. Means the disaccharides, we are talking about maltose, sucrose and lactose here. For all of them, maltose, sucrose and lactose, the formula will be C12H22O11. Okay. Now if the formula of glucose is C6H12O6, and if you want to make a molecule of maltose, in maltose they are made up of two glucose molecules. They are attached by alpha 1,4 glycosidic linkage. So if you are attaching two glucose molecules, 2 C6H12O6, Then if I ask what will be the formula of maltose, so without thinking you will answer that it will be C12H24 and O12, right? You will answer that it will be C12H24 and O12. You will add the atom numbers. But you have ignored one thing here. While one glucose molecule will make a glycosidic bond with another glucose molecule, there will be a dehydration from here. There will be a dehydration from here. One H2O molecule will escape away upon formation of alpha 1,4 glycosidic bond. That's why the formula will be, there will be minus H2O. The formula will be C12H22, 2 H atoms gone with water, and O11, 1 oxygen also gone. So this is a very tricky but easy question can be asked, right? Now when you talk about sucrose, sucrose is also disaccharide, is also called as table sugar and is formed by reaction between one glucose molecule and one fructose molecule. Okay, and one important thing that sucrose is also called as invert sugar. We know the another name is table sugar, and sucrose is also a non-reducing one, non-reducing sugar. Monosaccharides and disaccharides are called sugar. Who are sugar? Because monosaccharides and disaccharides, they taste sweet. That's why we call them sugar, monosaccharides and disaccharides. And most of them are reducing, but sucrose is one of the example of non-reducing disaccharide. Okay. Now you can comment to the box, into the comment box, why we consider sucrose as non-reducing sugar. Okay. Another disaccharide, lactose, present in milk, made up of one galactose molecule and one glucose molecule. Okay. Now we'll jump inside the polysaccharide. So polysaccharides are polymer of monosaccharides. You need multiple monosaccharides unit to form chain of polysaccharide. The bond is used to combine the molecules, monosaccharide molecules together, that is glycosidic bond. We'll talk about different types of glycosidic bond. There will be alpha 1,4 glycosidic bond, there will be alpha 1,6 glycosidic bond, there will be beta 1,4 glycosidic bond, there will be alpha 1,2 glycosidic bond. So there will be different types of glycosidic bond we need to talk about. There will be N-type glycosidic bond, there will be O-type glycosidic bond, right? And mostly these polysaccharide molecules are insoluble in water, and the important thing, the statement based or assertion reason based question can be asked that they have a reducing end and a non-reducing end, right? And where you will get reducing end? At left hand side you will get the non-reducing end, and the right hand side you will get the reducing end. So trick is like that. For right hand side, R for R, right hand side of polysaccharide chain has a reducing end, RH is for reducing end, and LH is for non-reducing end. R for R, RH is for reducing end. Okay, great. Now we'll learn how to form a polymer of glucose, polymer of fructose in some progressing slides. Okay, one more important thing, polysaccharides are non-reducing in nature, though polysaccharides have one reducing end, that is RHS, and one non-reducing end, but the total molecule is considered as non-reducing. Why it is so? Because only one end is reducing. Which sugars are called reducing sugar? If you treat them with failing solution or toluene solution, if they have the capability to reduce the copper ion, right, and they will exhibit change in color, so we call that sugar is a reducing sugar. But if you are taking a long chain of polysaccharide, it has only one single reducing end, but rest all the parts, all the units are non-reducing in nature, so that cannot reduce much copper ion in the toluene solution or failing solution. So it is said to be the polysaccharides are non-reducing in nature, right? Because the whole molecule is non-reducing, behaving as non-reducing, but only one single end is behaving as reducing point. So that will not be creating any difference when it is going for reduction of copper ion in failing solution. Okay. So let us understand in a detailed manner. So this is a chain structure, this is a strange chain structure of polysaccharide. Suppose any polysaccharides you can talk about. So this is the non-reducing end, the left hand side LHS, and this is the reducing end RE, the right hand side. Okay. So this is the reducing end, that's why I have written reducing here. Now how you can identify which end will be or which part is responsible for the reduction? Okay. So you have to find out the hemiacetal or a hemiketal group. You have to find which group? This structure, a carbon with one valency is catching one oxygen atom, and with another valency it is catching hydroxide atom. So this particular group is called hemiacetal group. Now if you observe, if you can identify this particular group inside the structure of a sugar, either it's a linear structure or it's a cyclic structure, then obviously with a blind eye you can say that yes, this particular portion is reducing in nature. Now let us try to find that where in this long chain this hemiacetal group is present or absent. So if you talk about this non-reducing end, this particular structure, so this is a carbon. With this carbon there is one group attached, that is hydroxide group. You got one hydroxide, you need to find one oxygen attached with it. So with this carbon there is one carbon, there is another carbon, and there is one H. So there is no oxygen attached with it. For this carbon, here you will get one oxygen, here also you will get one oxygen, here you will get one carbon associated with it, here you will get one hydrogen associated with it. So for this carbon also we haven't found any hemiacetal group. But when we are talking about this particular carbon, this carbon has one oxygen, this particular carbon has one hydroxide. With this part it is attached with one carbon, with this part it is attached with one hydrogen. So we have to search for this particular arrangement, one OH and one O. We got our hemiacetal group here. So that's why with this particular end it can reduce a copper ion in solution. Okay. So that's why we are calling this end as a reducing end. Now as because this particular glucose molecules, they are busy in forming glycosidic bond with the preceding molecules, their reducing structure has abolished. So that's why though there is only one reducing end, but the whole structure is non-reducing and the whole polysaccharide molecule is acting as non-reducing molecule. Okay. Now it's time to classify polysaccharides. Polysaccharides are of two types, homopolysaccharides and heteropolysaccharides. Now what is the difference between homo and hetero? Homo means repetition. The monomers are similar. Okay, the repetitive monomers are similar in case of homopolysaccharides. In case of heteropolysaccharides, the repeating monomers are not similar, they are of dissimilar types. So that's why we call them heteropolysaccharides. So polysaccharides are of two types, homo and hetero. Yes, so that's why it's written here, similar types of monosaccharides are present into homopolysaccharides, but different types of monosaccharides are present into heteropolysaccharide. Now again, homopolysaccharides are of two types, structural homopolysaccharide, storage homopolysaccharide. When you talk about structural homopolysaccharides, examples are cellulose and chitin. Why they are called structural? Cellulose take part in cell wall formation, chitin take part in cell wall formation in fungi, and exoskeleton formation in arthropods. Okay. Now why starch, glycogen and inulin, they are called storage homopolysaccharides? As because starch, the storage of glucose in plants, glycogen, storage of glucose in animals, and inulin, storage of fructose in few root tubers like in dahlia plants and artichoke plants. Okay. Now heteropolysaccharide, let's talk about their examples. Heteropolysaccharides, the examples are given here like agarose, they are obtained from red algae Gracilaria gelidium. Pectin, structural part of plant cell wall. Hemicellulose, again a structural heteropolysaccharide. Hyaluronic acid, yes, help in structure, a structural heteropolysaccharide. Where you will get hyaluronic acid? You will get hyaluronic acid into the basement substance, into the ground substance or intercellular matrix part in animal tissue. GAGs, glucosaminoglycans, they are also examples of heteropolysaccharides. Okay. Mucopeptides, the cell wall materials of bacteria, they are also examples of heteropolysaccharides. So I have made a table here. Of all the important polysaccharides. So, we will discuss about cellulose, chitin, starch, glycogen, inulin, agarose in shortcut manner. So please follow this table. Cellulose is homopolysaccharide made up of beta D glucose, and they are attached to each other by beta 1,4 glycosidic bond. You have to understand, you have to remember the type of bond in case of polymerization, and they will form unbranched linear chain. If we talk about chitin, they are also homopolymer made up of beta N-acetyl glucosamine, and this beta NAG, beta N-acetyl glucosamine, they are attached by beta 1,4 glycosidic linkage. They are responsible for the formation of exoskeleton of arthropods as well as cell wall of fungi. If we talk about starch, they are also homopolysaccharide made up of alpha D glucose. See the difference between cellulose and starch. Cellulose is made up of beta D glucose, but starch is made up of alpha D glucose. Glycogen is also made up of alpha D glucose. Okay, now starch. In starch you will get two type of glycosidic bond, alpha 1,4 glycosidic bond as well as alpha 1,6 glycosidic bond. There are two forms of starch present into nature. One is amylose and one is amylopectin. Okay. When you talk about glycogen, that is also called animal starch. Here also you will get alpha 1,4 type of glycosidic bond and alpha 1,6 kind of glycosidic bond. The structural similarities are there in between starch and glycogen. They will also form branched chain like amylopectin of starch. When you are talking about inulin, read it properly. This is not insulin. This is inulin. That is also homopolymer made up of beta D fructose, and the bond is used here beta 1,2 type glycosidic bond. You will find inulin into dahlia and artichoke. Okay. When you are talking about agarose, in biotechnology lab for gel electrophoresis we use agarose gel, and that's a heteropolymer, and you will find agarose, the source of agarose, seaweeds as red algae. Yes. Now we'll learn in detail the structure, function and every aspect of glucose. Yes, cellulose. Cellulose is homopolymer of beta D glucose. Cellulose is homopolymer of beta D glucose. Cellulose will form unbranched linear chain. Cellulose forms unbranched linear chain, straight line, right? And the bond is used here, that is beta 1,4 glycosidic linkage. Now we will understand how this beta 1,4 glycosidic linkage is produced in between beta D glucose molecule. So here you can observe how a polymerization is happening. Okay, this is one beta D glucose, this is second beta D glucose, this is third beta D glucose. Now let's understand how this beta D glucose will polymerize together to form cellulose molecule. So this is beta position of glucose, a beta D glucose. So this is a beta D glucose. This is another beta D glucose we will draw. Now you have to form glycosidic bond between them. Now where to form glycosidic bond? So glycosidic bond will be there. This is the first glucose molecule and this is the second glucose molecule. So in between the first carbon of the glucose molecule and for the preceding on the next molecule, we'll be using first, second, third, fourth glucose molecule, fourth carbon. In between first carbon and fourth carbon, the bond will be produced, and both are beta glucose. So the bond is called as beta 1,4. Now let's form glycosidic linkage. To form glycosidic linkage, again you have to perform one dehydration. Now how to perform this dehydration? Here you have to eliminate out 2 hydrogen and 1 oxygen by forming water. So when you will eliminate out this. This oxygen, the remaining oxygen will form a glycosidic bond. Or glycosidic linkage. So that's how beta 1,4 glycosidic linkage or bond is produced. Now this is a structural homopolymer and help in structure of a plant cell wall. Okay. Now let's talk about the iodine test. So generally iodine test will be positive for starch, but for cellulose the iodine test will be negative, right? It's insoluble in water. Like if you take out a piece of paper or cotton and if you try to dissolve into a glass of water, that's not possible, right? So that's why we say this cellulose molecules are insoluble in water. Where these cellulose molecules are used for the formation of paper, we'll find them into cotton, inside cotton and the woods, the desk or benches you are sitting upon. Okay. And the most important thing you have to remember about them, they are the most abundant biomolecule in biosphere. In living world, the most abundant biomolecule is cellulose. Remember this. Again, do not get confused between collagen, rubisco. Collagen, most abundant protein in animals. Rubisco, most abundant protein in biosphere. Now, most abundant biomolecule, all the plants and trees, forest made up of cell, and the cell wall has cellulose. That's why we can consider it most abundant biomolecule in biosphere. Now, after cellulose, we will talk about starch. Again, starch is produced in plants. So, homopolymer of beta D glucose. Okay, starch is not a homopolymer of beta D glucose, alpha D glucose. Starch is homopolymer of alpha D glucose. Cellulose is for beta D glucose, starch is for alpha D glucose. Okay. Now, excess glucose is produced after photosynthesis in plants. Now, the glucose cannot be stored for a long time. So, what happens in your home? The excess cooked food, will you throw it out? No, you will store it inside your refrigerator. So, plant, as because they cannot store glucose for a long time, so they polymerize it in the form of starch. So, excess glucose in plants are stored in the form of starch. So, there are two forms of starch is available. One is amylose and another is amylopectin. Now, let us understand their difference. So, amylose, they are unbranched, you can observe here. So, they will also form this kind of helical structure. So, they are soluble in water. You can see this helical structure, this unbranched helix is of amylose, and the glucose alpha D glucose molecule will form a bond of alpha 1,4 glycosidic bond to form amylose molecule. They give positive result in case of iodine test. If you try putting iodine solution on amylose, the iodine molecules will get trapped in these pockets of this helix, and that will form starch iodide, and that will give dark blue or blue black color, that is called positive iodine test. Now, let us talk about amylopectin, another variety of starch. Now, they are branched, unlike amylose, they are branched. Here branch formation can be seen here. Right, and they are insoluble. So, that is the difference between amylose and amylopectin. Amylose is soluble in water, but amylopectin is insoluble in water. Now, when you talk about the linear chain formation, just like the amylose, you will find alpha 1,4 glycosidic linkage, but wherever there will be branch chain formation, you have to introduce a new kind of bond, that is beta 1,6 glycosidic bond for branch formation. Remember this. So for linear chain, alpha 1,4 bond is enough. Now whenever you want to form a branch chain in that particular structure, the branch formation point, you need to introduce beta 1,6 glycosidic bond. Okay. In iodine test, it won't be producing purely blue black color, because it cannot trap a lot amount of iodine, but with this branch pocketing it can trap few of the iodine molecules, so it will produce some reddish brown color in iodine test. Now let us understand how alpha 1,4 glycosidic bonds are produced and how alpha 1,6 glycosidic bonds are produced. Alpha D glucose will form glycosidic bond with another alpha D glucose, right? Okay. So this is a linear chain, and here I am going to form a branched chain. So first let us introduce the alpha 1,4 glycosidic bond. So alpha 1,4 is between the first carbon and fourth carbon, in between the first carbon and fourth carbon. So here there will be dehydration, here there will be dehydration, and there will be formation of alpha 1,4 in between them. When the H2O will go out, the oxygen molecule will form a linkage in between this alpha glucose first carbon and this alpha glucose fourth carbon, right? But how to form alpha 1,6? So this is the first carbon, this is the sixth carbon, right? Here there will be formation of one glycosidic linkage. So this is how the alpha 1,6 glycosidic linkage will form upon another dehydration reaction. So the bond will be produced here, we call it alpha 1,6. This is alpha glucose, this is alpha glucose, this is first carbon, this is sixth carbon. So this is alpha 1,6, but these are alpha 1,4. This is alpha 1,4. So I think done, we understood how alpha 1,4 and alpha alpha 1,6 glycosidic bonds are produced. A mistake was there. Alpha 1,4 and alpha 1,6 glycosidic bond will be finding into the structure of amylopectin. Now let us talk about chitin. Now we will understand the structure of chitin. So chitin is the second most abundant biomolecule in biosphere, second most abundant biomolecule in biosphere. Is homopolymer of beta N-acetyl glucosamine. N-acetyl glucosamine, we call it NAG, and found in cell wall of fungi, cell exoskeleton of arthropods, like exoskeleton of cockroach. That's why when you touch cockroach it feels quite hard. We have endoskeleton inside our body that is made up of bone and cartilage, but arthropods, they don't have endoskeleton, they have exoskeleton which is made up of chitin. Okay. Apart from arthropods, you can get chitin in few other phylums as well. You can get chitin in few annelids, right? Okay. So let us move forward. The question can be asked, chitin is a homopolymer or heteropolymer? So chitin is homopolymer. Homopolymer of what? Homopolymer of N-acetyl glucosamine. So this is the structure of N-acetyl glucosamine. Already we have studied the structure. Yes. Now let us talk about the structure of glycogen. Glycogen is also called as animal starch. The structural similarities are there in between glycogen and amylopectin, that is a starch. It is homopolymer of alpha D glucose. Glycogen is homopolymer of alpha D glucose, and you will find glycogen inside our liver and muscles, sometime in our adipocyte cell or fat cells as well. Okay. Glycogens, the polymer structure will form branched chain like this. Branched chain is seen, and for iodine test it will give again the similar kind of test result whatever we got when we are checking the amylopectin. The red brown color will get in iodine test. Yes, bonds are also similar. When you are talking about the linear chain, the bond will be alpha 1,4 type for linear chain formation, and whenever we are talking about branch formation, we have to introduce alpha 1,6 glycosidic bond. Alpha 1,6 in previous slide, it was my mispronunciation, you can say. I don't know how I said beta 1,6, it should be alpha 1,6 in this particular slide. So again I am sorry for that. For the structure of amylopectin, it's for straight or linear chain alpha 1,4 glycosidic, and for branched chain it's alpha 1,6 glycosidic bond. Here you can observe obviously the glucose are alpha glucose, so there is no question the beta bond will be produced. Okay. Now let us come to another polymer of fructose, a polysaccharide. There is also again homopolymer of beta D fructose. It is found in root tuber. If you observe dahlia plant, they have a very beautiful flower. So root will produce tuber like structure for storage of fructose. So these structures are called root tubers, and they will store fructose as inulin polymer in which plant? Dahlia plant and artichoke plant. Okay, one more important thing is written here. They're also used for kidney function test. Okay, now for glomerular filtration rate test, GFR test, you can use inulin. Inulin in nanogram amount is injected into the patient's blood circulation or blood circulation of a person. Then in a span of time within 10 years you have to collect the urine. So if you are healthy, your kidney will filter out 100% of inulin. That is the significance your kidneys are working well. But if your nephrons, basically kidneys are made up of nephrons and they are filtering your blood, and if your nephrons are unable to filter out the 100% inulin, whatever the doctor had injected into the bloodstream, then there is some problem in the glomerular and Bowman's capsule junction, right. So that is a test of glomerulonephritis, sometime for kidney failure. Okay. Now I will talk about some heterocyclic ring, as NCERT is saying about heterocyclic rings. Yes. So if ring has elements other than carbon, then that ring is called heterocyclic ring. So here you have to identify which ring is heterocyclic and which ring is homocyclic. So if you observe this particular ring here, that has all the carbon atoms present at this corner of this ring. So you can say this is a homocyclic ring. If you observe these rings, you will get presence of oxygen inside the ring, oxygen atom. So these are heterocyclic. Obviously this is a pyranose ring, this is a furanose ring, you know why. And again the structures of pyrimidine and purine, the structure of nitrogenous bases. This is purine, this is pyrimidine. Remember, these are nitrogenous bases, purine and pyrimidine. Purine is a small word, pyrimidine is a long word, but the structure is vice versa, opposite, exactly opposite. The smaller word purine has a large structure, a complex structure. The large word pyrimidine has kind of simpler structure. So two rings are present into the structure of purine. This is the structure of purine, and obviously you can see that existence of nitrogen inside the ring. Existence of nitrogen inside the ring. Nitrogen is present at first position of ring, third position of ring, seventh position of ring, and ninth position of purine. For pyrimidine, nitrogens are present at first position and third position of ring. So what is the definition? If ring has elements other than carbon dioxide, obviously if nitrogen is present, that's these are also examples of heterocyclic ring. Okay, now we'll study about nucleic acids. So nucleic acids means DNA and RNA, deoxyribonucleic acid and ribonucleic acid. Now how to extract nucleic acid? First you have to take a cell, you have to take out the cellular fraction. You have inside the nucleus you will get chromatin fibers, and during the time of cell division this chromatin fibers will assort such a way in a proper manner. We will calling these densely stained bodies chromosome, chromaplasma, colored bodies present inside the nucleus. Basically, they are arranged version of chromatin fibers only. Now, inside the chromosomes, if you observe the chromatids, the arms, these chromatids are made up of a coil structure of DNA and proteins. You call it nucleosome model. And if you decoil it, if you eliminate out all the histone and non-histone proteins, you will get the structure of DNA. So, they are double stranded structure, and when you talk about RNA, they are single stranded structure. Now, how this DNA and RNA are made up of? We need to talk about, because DNA and RNA, these nucleic acids, they are polymer, they are macromolecular structure. So, we need to talk about their monomeric units. So, monomers of nucleic acids, DNA and RNA, are called nucleotides. Remember it. Monomers of nucleic acids are called nucleotide. Now, how you can form nucleotide? Nucleotides are formed by combining nucleoside with phosphoric acid. Nucleoside with phosphoric acid. Now, what do you mean by nucleoside, sir? You can form nucleoside. For nucleoside, you need pentose sugar, either it's ribose or you need deoxyribose sugar. If you add it with nitrogenous base, then you will get nucleoside, right? So, to form DNA or RNA or any nucleic acids, you need three components. You need pentose sugar, you need nitrogen base, and you need phosphoric acid. And when you combine them together, you call them nucleotide. If you combine only pentose sugar and nitrogen base, we call them nucleoside. So, that is why nucleotides can also be called as alternatively as nucleoside plus phosphoric acid. So, when we are talking about the formation of nucleotides, first we have to understand the three basic components which are required, that is, one is pentose sugar, another nitrogenous base, and obviously the phosphoric acid. Okay. Now, why we are calling these particular sugars as pentose sugar? So, the meaning of pentose is 5 carbon sugars. Basically, they are cyclic structure or ring structure, and there are two kinds of pentose sugar we will be talking about. One is ribose sugar and another is deoxyribose. So, both of them have 5 carbon, that is why we are calling them pentose sugar. Now, what is the difference between ribose and deoxyribose? Obviously, by the name you can guess that ribose sugar will be used for RNA and deoxyribose sugar will be used for DNA. Okay. Now, you can see the formula is written here. For ribose sugar, it's C5H10 and O5, and for deoxyribose sugar, there will be deletion of one oxygen. So, when one oxygen is deleted from the second carbon position, so the formula will be C5H10O4. Here you can observe that for ribose it's O5, but for deoxyribose it's O4. Now, let's talk about the structure of them. Okay. So, basically we have to draw what kind of ring. So, you know already we had discussed that this kind of rings are called Furanose ring, because inside the ring you will get 4 carbons and the 5th carbon is present here with CH2OH. So, this is the first carbon position, second carbon position, third carbon position. First carbon position, second carbon position, third carbon position, fourth carbon position and 5th carbon position. Now, you have to tag the functional groups with them. Okay. So, this is the ribose sugar, here there will be one hydroxide group, another hydroxide group, another hydroxide group. So we got the structure of ribose sugar. So in here opposite position you have to put hydrogen here. Now you can count the total number of carbon. C5, already 5 carbons are there. H10. So let's count the number of hydrogen. 1, 2, 3, 4, 5, 6, 7, 8, 9 and 10. O5. 1, 2, 3, 4 and 5. So it's done, it's satisfied. The structure and the formula is satisfied. So this is the ribose sugar. Now I am drawing deoxyribose sugar. Please focus on the second carbon position. Yes. So this is the structure of deoxyribose sugar. Again another furanose ring. Now let's fulfill all the vacant positions. First carbon, second carbon, third carbon, fourth carbon, fifth carbon. Okay, outside the furanose ring. Here there will be hydroxide, hydrogen, 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 hydroxide, hydrogen and hydrogen. Now as I said that you have to focus on the second carbon position. Now you can clearly distinguish in between them. The second carbon position, the group here you can observe that is hydroxide group, and here the group will be only hydrogen. So that is the difference, because one oxygen has gone already. That's why from ribose to deleted oxygen or deoxyribose sugar. Right, now we have to talk about nitrogen bases. Nitrogen bases. So there are two kinds of nitrogen bases required, and one is purine, another is pyrimidine. So purine base, as I said, purine base and pyrimidine base. Now the trick is there. If I give you these two particular structure, now I will ask you, please identify that which is purine and which is pyrimidine. Now you have to observe the structure and their complexity. For purine you can observe there are two rings are present, and for pyrimidine there is only one ring. So there are more letters. So pyrimidine is a longer word, but purine is a smaller word. But the structure is more complex for purine in compared to pyrimidine. Right? For purine there are two rings, for pyrimidine there is only one ring. Now there are two purines, adenine and guanine. Adenine is expressed as the letter A, and the symbol of guanine will be the letter capital G. Okay, now while we will be talking about pyrimidine, we have to talk about three pyrimidine nitrogen base, cytosine, thymine and uracil. Cytosine, C for cytosine, T for thymine and U for uracil. Okay. Now where we will be using adenine, guanine, cytosine, thymine and uracil as nitrogen base? Remember, adenine and guanine both are used for DNA as well as RNA. Both are required for DNA as well as RNA. Cytosine, again, it's required for DNA as well as RNA. Thymine required only for DNA, and uracil required only for RNA. Right? So if I say the nitrogenous bases which are required for DNA formation, then you will answer, it will be A, T, G and C. And if I ask you what are the nitrogenous bases required for the formation of RNA, then your answer will be A, U, G, C. So that is the difference. For DNA, thymine, for RNA, uracil. Got it? Now I will be differentiating between purine and pyrimidine structure. Now you have to check for the position of nitrogen. Okay, so it's basically both are heterocyclic ring. So the position of nitrogen will be like this in purine. The nitrogen is present inside the ring in the first position, the third position inside the ring, the seventh position inside the ring, and the ninth position inside the ring. Now for pyrimidine, the placing of nitrogen will be like this: first position and third position inside the ring. You need to remember this. Okay, that will help you when you will be forming nucleosides as well as the nucleotides. Okay, now the third component is needed, that is phosphoric acid, and this is the component which will be making the DNA or RNA molecules negatively charged as well as acidic in nature. We said DNA and RNA, they are nucleic acids, right? Deoxyribonucleic acid for DNA and ribonucleic acid for RNA. So this acid terminology, the acid nature, the acidity, we will be getting from this particular structure, from this particular group. The group is called phosphate group, and we will be deriving it from the phosphoric acid. Now let's talk about the structure of phosphoric acid. The chemical formula of phosphoric acid is H3PO4. H3PO4. Okay. Now there will be a central phosphate, and one oxygen is associated with the phosphate group with the double bond, and 3 single bonds are there, and 3 hydroxide molecules are associated with it, right? So that is the structural aspect of phosphoric acid. Now whenever we will be forming phosphoester bond, whenever we will be tagging phosphoric acid with some other molecule, it will be forming phosphoester bond. By the process of dehydration, we have to utilize this hydrogen, and this hydroxide group can donate the H plus ion, and it will become O minus, and that will behave like acid. What is acid? The chemical compound which has capacity to donate hydrogen, that is called acid. So here these hydrogens can be donated. Okay. Now let's talk about the formation of nucleotides. So as we already know that how nucleosides can be formed, for that we need a pentose sugar, and we have to tag nitrogen base with that. Now this pentose sugar can be ribose sugar, the pentose sugar can be deoxyribose sugar. Okay. So suppose we are taking, you can take an example of any of the nitrogen base. You can take adenine, you can take guanine. Now where is the position, like with what particular number of carbon, with which point you have to tag the nitrogen base? For that generalized formula is there. So basically that will be the first carbon of pentose sugar, PS for pentose sugar, will be associated with ninth nitrogen of purine. Okay. Similarly, first carbon of pentose sugar will be associated with first nitrogen of pyrimidine. You have to remember this. And the bond in between the pentose sugar and the nitrogenous base will be glycosidic bond. Glycosidic bond. The bond between pentose sugar and nitrogenous base, that will be glycosidic bond. You need to remember this, the question can be asked. Yes. Now I am taking ribose sugar first. Then I will take deoxyribose sugar. So this is ribose sugar, this is deoxyribose sugar. This is ribose sugar, this is deoxyribose sugar. This is the second carbon position. Now you know why it's so. Okay, now we have to tag what? We have to tag nitrogenous base. Suppose we are tagging adenine. So in the first carbon position, we have to combine the adenine with the glycosidic bond, and here also I am adenine. So when the sugar nature of pentose sugar is changing, the name will also change. So for this nucleoside, we will be calling it adenosine. We will name it adenosine. And for this, where adenine base is attached with the deoxyribose sugar, we are naming it deoxyadenosine. So that is the difference between when we are using pentose, when we are using ribose sugar and when we are using deoxyribose sugar. I think you got it. Now, what are the different types of other types of nucleosides can be formed? Let's make a list. Okay. So whenever we will be using ribose sugar with nitrogenous base, we can form adenosine. When G will be tagged, that will be guanosine. When C will be tagged, that will be called cytidine or cytidine. So when you are adding purine, it's sin. When you are adding pyrimidine, it's dine or dine. So adenosine, guanosine, cytidine, thymidine, and uridine. When you are tagging deoxyribose sugar, it will be deoxyadenosine. Deoxyguanosine, deoxycytidine, deoxythymidine, and deoxyuridine. Okay. Now there is a problem. And what's the problem is? The problem is that you cannot use all of them, for DNA or RNA formation. Like I'm giving you that instance, right? So adenine can be used for RNA, deoxyadenosine can be used for DNA formation. Guanosine can be used for RNA, deoxyguanosine can be used for DNA only. Cytidine and deoxycytidine, one is for RNA, another is for DNA. But there is no use of thymidine for RNA. There is no use of thymidine for RNA, because RNA will be utilizing for RNA formation, you need formation of uridine, not thymidine. Similarly, in case of DNA formation, there will be no use of deoxyuridine, so that is again useless for us, right? So technically, formation of deoxyuridine is possible in artificial synthesis manner, but inside our body there will not be formation of deoxyuridine, and there will be no formation of thymidine. There will be formation of only deoxythymidine. I think you got it. Now we are moving forward to the next slide, that is formation of nucleotides. Nucleotides. So for nucleotide formation, we already know that we need nucleoside plus phosphoric acid. Okay. Now what is the position? You need to tag phosphoric acid. Now it's always the 5th carbon position. Now again I will draw one ribose sugar and one deoxyribose sugar. Suppose this is the ribose and I am making a copy. Here this is the ribose, here this is the deoxyribose and the carbon is second carbon position. The very first carbon position you have to tag any of the nitrogenous base, like I have added adenine. So now this molecule is adenosine and this molecule is deoxyadenosine. So both of them are nucleotides or deoxynucleotides, a nucleoside and deoxynucleoside, nucleoside and deoxynucleoside. Now we have to convert it into nucleotides and deoxynucleotide by adding phosphate group or phosphoric acid. So where you can tag phosphate group? In the fifth carbon position, and the fifth carbon is like this, CH2OH, right? This is the fifth carbon. Here you have to tag phosphate group. So there will be a esterification bond formation and we will be calling that bond as phosphoester bond. Now, when this phosphoester bond will be formed, the configuration will look like something look like this. Now this is the phosphoester bond formation. Now this molecule can be called as adenosine monophosphate or adenylic acid. You can also call it this because this is the adenosine and this is the phosphoric acid. So it can be also called as adenosine monophosphate. There is only one phosphate present into this molecule. So it also can be called as AMP, adenosine monophosphate, and this is how you can name nucleotides. Now what about deoxynucleotides? Similarly, in this place you have to tag the phosphoric acid molecule with the fifth carbon position like this, and now we got DAMP, deoxyadenosine monophosphate or deoxyadenylic acid. Got it? Now again, by adding more phosphate, you can make them into ATP or DATP. If you add 2 phosphoric acid, then it will be ADP, adenosine diphosphate. If you add one more phosphoric acid, 3 phosphoric acid, so it will become ATP, adenosine triphosphate. Similarly, DAMP can be converted into DADP and DATP. So how ATP is made? Let's see the structure. This is adenosine. Now first phosphate group, next we have to add second phosphate group with this. You have to add third phosphate group with this. Here we get the structure of ATP, adenosine triphosphate, right? Now we don't have to talk about adenosine triphosphate formation. We have to talk about only this nucleotide formation, and this bond is glycosidic bond. And this bond is phosphoester bond. This bond is phosphoester bond. So these are the different types of bonds you need to form a molecule of nucleotide or deoxynucleotide. Now, what can be the different types of nucleotides and deoxynucleotides? For RNA formation, you need adenylic acid, guanylic acid, cytidyllic acid, and you need uridylic acid. You don't need thymidyllic acid. So these are different nucleotides which you are going to use for the formation of polynucleotide, that is RNA, ribonucleic acid. And if you add deoxy with them, deoxyadenylic acid, deoxyguanylic acid, deoxycytidyllic acid, deoxythymidyllic acid, deoxythymidyllic, there will be no deoxyuridylic acid, you are going to form DNA molecule. Yes. So now we will see how you can form RNA or DNA molecule or polynucleotide molecule by adding them. Yes. So basically we will study formation of simple DNA. So in about DNA you are going to learn in detail in genetics chapter. So here the basic diagrammatic representation and structural representation will be there. Now let's form the polynucleotide by adding many NMPs, deoxynucleotides. By the process of polymerization of multiple deoxynucleotides, we will be forming polynucleotide, that is one chain of DNA, and when you combine the two antiparallel chains of polynucleotides, there will be formation of one complete DNA molecule, that is double stranded or double helical structure of DNA. So this is a deoxynucleotide. Suppose you are putting G here, so it will be a deoxyguanylic acid, right? Now we can copy this to form a polynucleotide chain. Like this. Now we have to tag them vertically. Now how this nucleotide will be connected, will form a bond with nucleotide which is present below to that. So the position will follow like this, with third carbon position of a nucleotide or deoxynucleotide, there will be bonding of fifth carbon position of the nucleotide or deoxynucleotide which is present just below to that. So for a chain formation, the linkage, phosphodiester linkage will be there, phosphodiester bond formation will be there, and will be following the direction or the molecules, the center of molecule can be utilized, that is for a single nucleotide or deoxynucleotide we will be utilizing, will be focusing on the third carbon, and for the below one we will be focusing on the fifth carbon, the phosphate which is associated with the fifth carbon with that. Now we will be forming phosphodiester bond here. Okay. So for again formation of phosphodiester bond, there needs dehydration. So upon dehydration, here there will be dehydration, here there will be dehydration, here also there will be dehydration. There will be formation of phosphoester bond, right? Now this chain is formed with the process of polymerization, and the bond is utilized here. The bond is here, that is called phosphodiester bond. Phosphodiester bond, because the phosphate group is attached with this fifth carbon with one single ester, and this phosphate group is attached with the third carbon of the nucleotide or the deoxynucleotide present above to that with another ester bond. So that's why we are calling this phosphodiester. Phosphodiester bond will connect one nucleotide to another one, or one deoxynucleotide to another deoxynucleotide. So multiple phosphodiester bonds we are going to use to form a long chain of polynucleotide. So this is one chain is formed. Now why we call it 5' to 3'? If you observe the very first nucleotide here and the last nucleotide here, what you can observe here, for the very first nucleotide, this fifth carbon is unutilized. It's not utilized for formation of phosphodiester bond. So we are naming this end as 5' end, and when you are observing the last nucleotide, you can observe here the third carbon position is unutilized because no phosphodiester bond is formed here. So we are naming it as 3' end. That's why one chain of DNA is named as 5' to 3', because of the positioning of the free carbons. One is fifth carbon and another is third carbon. Okay. Now we need such two chains, two anti-parallel chain to configure to form a DNA molecule. So these are the different building blocks. Is one base with attached with another base, so that is called base pairing. Now how this base pairing gonna take place? There will be formation of hydrogen bonds in between the nitrogenous bases of antiparallel chains, and according to the Chargaff rule, adenine will form double hydrogen bond with guanine, with thymine, and guanine will form triple hydrogen bond with cytosine. Right? Who was the discoverer? Erwin Chargaff, one Russian scientist. Yes. So this is how DNA double helix will be formed, and we'll be calculating the length of DNA by counting the number of base pairs. So if you observe this particular chain, how many nucleotides are there? 1, 2, 3, 4 nucleotides are there. So this is, it can be said as 4 base long. So similarly, if you attach to an antiparallel chain, this DNA molecule gonna become 4 base pair DNA. For this DNA molecule, how many base pairs are present? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. So this DNA molecule is a 13 base pair DNA. Now what is the width of DNA? What kind of DNA is it? Is a bDNA or aDNA or zDNA? What is the length of one turn? Everything we are going to learn into the genetics chapter. not in this chapter we will learn about all these things, right? Okay, so let's talk about the concluding part of DNA and RNA or nucleic acids. So already we have learned that how two antiparallel chains are formed by combining two nucleotide, two polynucleotide chains by formation of hydrogen bond between the nitrogenous bases. Always adenine will form double hydrogen bond with thymine. You can observe here G is forming triple hydrogen bond with cytosine or C. So always there is one purine and one pyrimidine. Always there is one purine and one pyrimidine. A is purine, T is pyrimidine, G is purine, C is pyrimidine. There won't be any bond formation between A and G, T and C. So purine-purine bond or pyrimidine-pyrimidine bond formation is not possible to maintain the even distance between the antiparallel chains. There is 20 angstrom, right, or 2 nanometer, the distance between two antiparallel chains, that will be 20 angstrom or 2 nanometer. Okay, so if you use both the purines, it will be too much wider, and if you use both the pyrimidines, it will be too much narrower. So to maintain the even distance, we will be utilizing one purine molecule and one pyrimidine molecule for the bonding purpose. Yes. Okay. So if you observe this particular DNA molecule, how many base pairs are present here? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So basically that's a 10 base pair DNA, 10 base pair DNA, and few rules are there you have to follow. First rule, the number of A, you can see the box, this is not the this box is not denoting towards the hydrogen bond. This is the box which telling about us about the hydrogen bond, but this is the box which is telling us in a DNA molecule the number of adenine will be equal to the number of thymine, the number of guanine will be equal to the number of cytosine. So similarly we can say that the number of purine will be equal to the number of pyrimidine, and the summation of adenine and guanine will be equal to the summation of thymine and cytosine. So these are very 3 short formulas you have to practice again and again. Many small tricky questions can be asked from these particular things. Okay. Now few really tricky questions can be asked. If you are given with one linear chain of RNA and you will be asked, tell me how many nucleotides are present. It's very easy to calculate, right? It's very easy to calculate the number of nucleotides on a long RNA chain, and the question can be asked that how many phosphoester and how many phosphodiester bond present inside one single chain of RNA. So if suppose this is the RNA chain and the bases are total, how many number? 1, 2, 3, 4, 5, 6, 7 and 8 bases are present, 8 nucleotides are present here to form this RNA chain. So how many phosphodiester bond will be there? In between first and second nucleotide there will be one phosphodiester bond. In between second and third there will be another phosphodiester bond. So the number of phosphodiester bond will be 1, 2, 3, 4, 5, 6, 7. Okay. So for RNA it's very easy to calculate the number of phosphodiester bond because only one chain is there. You just have to calculate the total nucleotides sequence wise, and you have to minus 1 from them, right? If the RNA chain is made up of 1050 nucleotide, then the number of phosphodiester bond will be there, that is 1050 minus 1, that is 1049 number of phosphodiester bond will be there. Okay. Now how many phosphoester bond will be there now? for calculating phosphodiester bond, one phosphodiester bond equals to 2 phosphoester linkage, right? One phosphodiester bond equals to 2 phosphoester linkage. So if you are asked to calculate the number of phosphoester linkage, so you have to double the number of phosphodiester, and for a molecule you have to add plus 1 with that. Why plus 1? Because at this 5 dash end there will be a free phosphoric acid molecule with only one phosphoester bond formation with the 5th carbon. So that's why please think twice before marking the correct answer. The question is asking about phosphodiester bond or phosphoester bond. Always add plus 1 while you are calculating the number of phosphoester bond upon multiplying with 2 with phosphodiester bond. Okay. Now for DNA, if N is the base pair of DNA, suppose it can be 10 base pair, then N will be 10. If the DNA is of 50 base pair, the N will be of 50. Now the question is asked, you have to calculate the phosphodiester bond, and in shortcut we will be signalify it with small d, that is the number of phosphodiester bond. The formula is given here, 2 into n minus 1. This is the formula. So if you are asked that if a DNA molecule is of 50 base pair, then how many phosphodiester bond will be present? So if you calculate by following this formula, it will be like this: 2 into 50 minus 1, it will be 2 into 49, and that is 98. So for a DNA with 50 base pair, DNA of 50 base pair long molecule, the number of phosphodiester bond you are gonna get, that is 98. Now another question is here, you have to calculate the number of phosphoester linkage into that same DNA, right? Here in this case the formula goes 2D plus 2. Now D means the total number of phosphodiester, right? So the phosphoester, the signal is here like small e, so you have to calculate small e. In case of a DNA which has 50 base pair, you have to calculate small e. So how to calculate that? You have to follow this particular formula. So 2D was 98 plus 2. The number you are getting, that will be the total number of phosphoester bond. So that's how these are the very easy but tricky simpler mathematical formulas, right? Very easy mathematical formulas. Even these questions, if you understand once, can be solved by a student who is studying in class 5 or 6, right? Okay, so don't worry about that. This kind of easy questions can be asked. Now the last topic of this chapter, of this particular biomolecule part, that is metabolites. So what is the definition of metabolites? Metabolites means, first you have to understand what is metabolism. So inside our each and every cell, inside our cells, every time continuously thousands of chemical reactions are going on. Now few of them are constructive, few of them are destructive. Now all these combinedly, all these chemical reactions, we are calling them metabolic activities or metabolism, and that metabolism is one of the defining feature of all the life form. As because we are performing metabolism inside our cells, we are alive. As soon as the metabolisms will stop or arrested, then we will die, correct? Now as I said, few of the metabolisms, they are making our body, and few of the metabolism, they are breaking our body. Few are constructive, few are destructive. Now the constructive metabolic processes are called anabolism, and the destructive metabolic process are called catabolism. catabolism for destruction, anabolism for something when your cells are getting remodeled, renovated. So basically during the time of anabolism your cell will gain dry weight, and during the time of catabolism your cells will lose weight. So one of the very good examples of anabolism is photosynthesis. So if you observe photosynthesis, by combining smaller molecules like carbon dioxide and water, autotrophic organisms are forming a large molecule, that is glucose. So that is adding dry weight of plant cell. Whereas while we are respiring, so the oxygen we are inhaling, that is getting inside your body, inside your cells by the process of diffusion, and that is responsible for oxidation or destruction of stored glucose molecules inside our cells. So for the process of respiration, oxygen is destroying glucose molecules, burning or oxidizing glucose molecules inside the cell, and by breaking large molecule of glucose we are getting carbon dioxide and water. So this kind of destructive process we are calling them catabolism. Okay, but either this metabolic process are anabolism or catabolism, always a few products are formed as a process of metabolic reactions, and those products are called metabolites. So simple, very simple, products of metabolism are called metabolites. But we have to understand the difference between primary metabolite and secondary metabolite. Now what are they? Primary and secondary metabolite. So it's very easy to understand, though you people are always in confusion. See, primary metabolites are produced by all members. Primary metabolites are produced by all members of individuals of a kingdom. Like if I'm talking about chlorophyll, you'll be finding this chlorophyll in all members of kingdom Plantae. If I'm talking about glucose, it will be produced by all members of kingdom Animalia, right? So that will be a primary metabolite. So primary metabolites are produced by all members, at least for one kingdom. Okay. Now when we study about these primary metabolites, for these metabolic reactions inside our body or metabolic pathways inside our body, we will be finding they have proper role to play. So they have identifiable function in the growth and development of our body. That is the general physiology. Okay. But in contrast, when we are talking about secondary metabolites, they are produced by some plants, fungi and microbes. We are not talking about us, animals. So secondary metabolites are for plants, fungi and microbes. They are product of these organisms other than animals. But again, the secondary metabolites are not produced by all plants, all fungi, all microbes are produced by few plants, few special fungi, few microbes, and they don't have identifiable function into the growth and development of plant, fungi or microbes. Like we'll be getting antibiotics from fungi or bacteria, right? For one example, the discovery of this the great antibiotic penicillin by Dr. Alexander Fleming from the fungi called Penicillium notatum, right? So penicillin is an antibiotic, the product of a fungi, but there is no direct function in the physiological growth or development of that fungi called Penicillium notatum. That's why we can say that the organism which is producing a particular secondary metabolite, there is no direct function of that into their body, right? So that's why we are calling them secondary metabolite. But few of the secondary metabolites are useful for us. They are useful for human welfare, and few of the secondary metabolites are really important for this ecology. Like for examples, spices or antibiotics produced by fungi and microbes are useful for us, are life-saving drugs for us, right? And flavonoids, special secondary metabolites produced by plants, have a great ecological importance. They are saving the plants against the ultraviolet radiation, right? So, for primary metabolites, all the primary metabolites have direct role to play into the metabolic pathways of our body. But for secondary metabolite, firstly, they won't be produced by all the members. They are produced by only few organisms. They are also not animals. There will be plants, fungi and microbes, and we don't know about their proper role or proper function into their body. Now, it's time to understand their examples. Okay. So, the first example is given here, glucose. That is the respiratory, for respiration, the food material that is required for almost all the cells, that is glucose. Ribose, again a pentose sugar, a monosaccharide. Lipids like cholesterol, fatty acid, glycerol. For structural aspect of cell, you can observe cholesterol in the cell membrane, you can observe phospholipids in the cell membrane. Amino acids, the nib, the monomers of proteins. Cholesterol, the precursor molecule of multiple vitamins like vitamin D, multiple hormones like androgens. Nucleotides, nitrogenous bases required for DNA and RNA formation. And chlorophyll, without chlorophyll plant cannot trap sunlight and perform photosynthesis. So, chlorophyll is a pigment. So, you know that chlorophyll is a pigment but acting as primary metabolite. But other pigments, when you talk about carotenoids, xanthophylls, right, those pigments are not considered as primary metabolite, they are secondary metabolite. Let us learn some examples of secondary metabolites. Rubber, gums, secondary metabolites. Spices, secondary metabolites. Antibiotics, secondary metabolites. Pigments other than chlorophyll, secondary metabolites. Scents, secondary metabolites. Cellulose, though you will be thinking that cellulose is the main cell wall material of plants, but when you observe the formation of a new plant cell, first there will be formation of primary cell wall, then secondary cell wall, then tertiary cell wall, middle lamella. So, those cellulose is giving structural strength of plant cell, but that is not directly involved into their growth and metabolism. Okay. Now, one table is given in NCERT, and learning this table is very much important for us, because you can expect sure short questions from this particular table of secondary metabolite. I can say the most important table of the chapter biomolecule. Okay. So, the table is saying about secondary metabolites. So, first we will talk about pigments. So, there are two pigments examples are given, that is carotenoids and anthocyanin. Pigments, carotenoids and anthocyanin. Now, with a great trick you can remember them. That is, a car is there and a pig is driving that car and an ant is sitting in that car. So, pig, car, ant. So, by this, pig means pigment, car means carotenoid, ant means anthocyanin. Okay. Another secondary metabolite, alkaloids. Morphine, codeines are alkaloids. One trick is there, alka codes more. So, nowadays every day they are coding, they are learning coding, right. So, alka, there is a girl, alka, she codes more. So, alkaloids for codeines and morphines. Okay. Now, terpenoids, they are used for essence or flavor formation, artificial food flavor, monoterpenes and diterpenes. So, you eat vanilla flavor ice cream, you eat orange flavor something which has orange flavor. So, this vanilla flavor, orange flavor, what do you think? They are extract of vanilla, they are extract of strawberry, they are extract of orange? No, they are basically these terpenoids. Essential oils, they used for cosmetics as antimicrobial agent. Example is lemongrass oil. So lemongrass oil is there, and essential oil also oil is there. So oil to oil you can easily memorize this. Now lectins. Examples of lectins are concanavalin A. So these concanavalins are used as biological response modifiers. They are used as anti-cancerous drugs, okay, and the shortcut trick is cola. You drink cola, right? Coca-Cola. So this cola, so co for concanavalin and la for lectins. Okay, L for lectins. Now drugs, there are few examples of drugs are given here. Curcumin is the extract of turmeric, which is used against Alzheimer disease. Vinblastine, tumor suppressor drug used against cancer. Okay, few toxins are there. Abrin, ricin. Again, these abrins and ricins are used for chemotherapy patients, right? So shortcut trick is toxic ab rice. And polymeric substances, very easy to remember, they are rubbers, gums and cellulose. They are polymers. Obviously you know about cellulose. Cellulose is polymer of beta D glucose. Okay. Now in the comment box you have to answer me. Even these questions are asked into NCERT. Please search and tell me, when you are talking about rubber and gums, they are polymers, but what are the monomers of rubbers and gums? Are they carbs, are they proteins, or are they lipids? What are they? Just comment in the given comment box. Okay. Now let's solve some important questions, and obviously very easily you can answer them. Let's test your knowledge. Which element is present negligible in human body? So you know the answer. The element is silicon. That is the second most abundant element in earth crust, but almost negligibly present into our body, that is much toxic for us. That is silicon. Okay. Next question, which of the following secondary metabolite is a polymeric substance? So the answer will be rubber. Yes. Next question, which of the following is the most abundant element present in human body? Most abundant element present in human body. The chart of CNR Rao, and the abundant element will be oxygen. Next question, elements that are found more abundant in human body than in earth crust. Chons, CHONS, chons or cons. Carbon, hydrogen, oxygen, nitrogen, sulfur. And Nakamag or Nakamagsi, sodium, silicon, calcium, magnesium, they are present more in earth crust in compared to human body, right? Next question, in a polysaccharide like glycogen, the right end and the left end are called. So when you are talking about the polymer of a polysaccharide, there are two ends. The right hand side, that is for the reducing end, and the left hand side, that is for non-reducing end. So the right end is reducing and the left end is non-reducing. Yes. Which of the following is acidic amino acid? Obviously glutamic acid, and another example was aspartic acid. There are only two acidic amino acids. Very easy to remember them. Glutamic, very easy to learn also, glutamic acid and aspartic acid. Three basic amino acid, arginine, histidine and lysine. Okay, following structure is related to which amino acid? So you can, the structure is given in NCERT as well. Which group is this? This is methyl group. So if this methyl group in case of, in the place of R group, so the amino acid is gonna be alanine. If there is hydroxymethyl, the amino acid is gonna be serine, right? If instead of CH3, if you get H here, then the amino acid is gonna be glycine. Okay, which of the following component is a component of simple lipid? Glycerol, It's a simple lipid. Fatty acids are also simple lipids. So our answer is both 1 and 3. Next question, lipids may be monoglyceride, one glycerol, one fatty acid, monoglyceride, diglyceride, one glycerol, two fatty acid, diglyceride, triglyceride, one glycerol molecules plus three fatty acid, triglyceride. So answer will be all of the above. Next question, following the structure is related to which compound? So the diagram of a nitrogenous base is given in NCERT, and this is a pyrimidine, and adenine is not a pyrimidine, it's a purine. Guanine is again a purine. So pyrimidines are here uracil and thymine. Now this is uracil or thymine. This is a uracil molecule. When you methylate it like this, then this will become thymine. Remember, this is very easy to understand. Methylation of uracil will give thymine. Next, DNA is acidic. Already we are discussing the same question. DNA is acidic because of this phosphoric acid. The phosphoric acid structure is like this. So due to these structures, phosphoric acid is acid, and that makes DNA acidic in nature. So with this we are ending the chapter of biomolecules. Again, the parts enzyme will be coming into another one shot video. Now for the timing, stay tuned in our channel of PW NEET English, and for today it's again thank you, and please solve the DPPs given in our PW app and let me know are you liking our one shot videos or not. And please, if you haven't subscribed our channel, subscribe it and press the bell icon for all the updates and notification. Till then, Jai Hind, bye bye.